undergraduate and graduate teaching. He has been a fellow of the Institute of Physics, London, England since 2004. So please, let's give him a warm welcome and take it away. Hello.
Thank you. I hope everyone can see my my screen. Yes? Can you?
No longer.
No.
Let's see. Okay, so let me make sure that I'm sharing my screen properly. Give me a moment to do that. Okay, here we go. And okay, now you can see it?
Yes.
Okay, great. All right. Okay, so the title of my talk is From Living Organisms to Living Machines. So let me give you an outline. I want to start by talking about what I think about as the borders between the living and non-living, which is relevant to our meeting. And I also want to talk about the great shapers of biological cells and organisms, evolution, development, plasticity. And to try to make this as concrete as possible, I'm going to do a thought experiment with you, and we will together think about something I'm going to call a cell machine. It will have, perhaps that's too grandiose, we'll see that it has certain limitations, but it may also get us thinking about how one would take these ideas and implement them for artificial devices. And in order to understand more about control for such a device, I will also talk a little bit about creating biologically inspired neural networks. And then I will briefly talk about some of the new directions. Where most of the people here are more focused on going from the artificial to the biological or being inspired by biology, but biologists are actually going in the sort of the opposite direction, trying to create artificial organisms. And so I'll briefly talk about synthetic biology and artificial life. And as we start to blur these borders and are successful in doing so, I think we're going to have to also deal with some of the social and ethical considerations that this raises. And then I will summarize and hopefully reach some interesting conclusions. If someone has a question as I talk, you're welcome to ask, but I will also hope to leave some time for questions afterwards. And my hope is to really, this is sort of a, as opposed to being a talk where I focus a lot on my own previous work, this is a talk where I'm hoping that I can get the whole group thinking about some of these fundamental questions with me. So our conference is called Living Machines, and it raises two very fundamental questions. The first question is, are living cells and organisms machines? Is that a fair way of describing them? And then, are there any human-created machines that are alive? Now, I recognize that these questions might seem almost too philosophical, but I hope to convince you during the course of the rest of this talk that these are actually useful questions to answer, and they both help us understand more about living things at the same time as they perhaps inspire us to create novel artificial devices. There's a very interesting and thoughtful essay that was recently published by Cornish, Bowden, and Cardenas, and they compare and contrast several possible theories. I'll just mention them, I won't go into them in detail. There's the work of Maturana and Varela in the 70s on autopoiesis, self-creation. There's the work of Eigen and Schuster on hypercycles. There's the work of Rosen on what he calls MR systems. There's the work on autocatalytic systems, and there's work of Ganti on what he called the chematon. And so they go through these, and I will say more about what they derive from all of them. But at the outset of their essay, they make some very important points, which I think I'll start to sort of set the framework. The first point they make is that no matter how one defines a machine, whether a very simple tool, such as an axe, a more complicated machine, such as an airplane, or even an entire factory, it remains true that the machine does not make itself or maintain itself. It needs the input of an external agency in order to do this. I think someone may need to mute themselves because I'm starting to get echoes. Roger, that may be you. Okay. And the second point they make is that living organisms are quite different from machines. They depend on their environments for material, food, and they release material, such as CO2, water, and other excreta into their environments. So they are thermodynamically open systems. That is, they take sources of energy, They maintain internally low entropy, high structure, but they release material that has much higher entropy, so thermodynamics is not violated. But they are organizationally closed systems. We'll say more about that in a little while. And all of the catalysts they need, that is all the structures internal to them that allow them to transform, for example, energy sources or materials that they themselves are manufacturing or receive from other cells, are closed systems. That is, they make and they maintain all of those catalysts internally. And all the catalysts they need, apart from some metal ions, they make themselves. Okay, so this in a certain sense throws down the gauntlet because it tells us that if we claim that something is a living machine, we have to argue that somehow it actually has some of the characteristics of living things. So this raises the question, what are the characteristics of living things? And one of the ways that people try to do this, you'll find this in textbooks, is that they list characteristics associated with living cells and with organisms. And it's essentially kind of a checklist. So living organisms and individual cells show internal organization. They have metabolism. That is, they have the network of processes that are capable of taking in certain inputs. I mentioned energy sources. They can transform one set of chemicals into other chemicals, and many of the things they transform the chemicals into are either needed for maintaining themselves or for their function. So for example, if a molecule is involved in generating motion, then there will be the universal energy currency, ATP, will be made available such that those energetic events can occur. So if you have, for example, molecules that can actually walk, the ATP will power that, or you'll have muscle molecules which can seize hold of one another and then move, giving rise to muscle contractions. All of these things are maintained by the internal metabolism. Another critical function is homeostasis. They maintain certain levels of certain chemicals at certain values, and they work either as individual cells or as organisms as whole to stay within certain ranges of acidity, temperature, pressure, and so forth. They're capable of growth, so they can make themselves larger, and they can reproduce. They can make copies of themselves. And finally, they're responsive to the environment. As things change around them, they can change appropriately to what's going on around them. So to try to show both the applications of these ideas, but some of their limitations, let's look at some examples. So if we do so, we'll see that the borders are a little bit fuzzy. So let's start with something that in principle should be very easy. So let's ask the question, is a rock alive? So if you actually look inside the structure of rocks, you see that they have highly organized crystalline regions. Think of a geode, for example. So that's something that actually is highly organized, and crystalline structures in general are highly organized. So that satisfies one aspect of the checklist. And within certain ranges, if you apply force to a rock, you throw it, or you step on it, you throw it into water, it maintains its integrity. In that sense, it has a kind of resistance to change, which you could refer to as a kind of resilience. But in terms of the other parts of the checklist, it certainly doesn't, in general, grow, reproduce, and so forth. So it doesn't satisfy all the aspects of the checklist, but it already shows you that using a checklist is problematic because other natural phenomena have some aspects that are clearly shared with other living things. Let's try a harder case. Is a virus alive? And I just want to say, I'm not going to take a stand on this. I'll point out the reasons why people say yes and people say no. But in the literature, the technical literature, will find that this is actually a debate. Some people say no and some people say yes. And the reason is, and unfortunately, because of our current situation, we are much more familiar with the effects of viruses on human health. So if you've ever seen pictures of viruses in detail, you see that they have highly organized internal structure. The particular one we've been dealing with has these rather beautiful looking spikes on it, which give its name corona. And internally, it also has complex structure, including the messenger RNA that it needs for making copies of itself. However, unlike other living things, you can actually make many numbers of viruses, and you can crystallize them as a powder, and you can put them on the shelf for long periods of time. And the reason you can do that is they can only reproduce by taking over the machinery of a cell. So for example, bacteriophage, they look like a lunar excursion module. They have these little legs. They can land on top of the cell. They can inject their material that hijacks the cell. The cell takes over, making all of these different components, and they self-assemble. The cell dies as a consequence, and it releases very large numbers of the viruses. And a critical aspect, which we are dealing with on a fairly regular basis, is that viruses make poor copies of themselves. So some of the duplicates are much worse than the original virus, and some are actually far better at, for example, evading the immune system or dealing with someone who's vaccinated. So they have this ability to change over time. Are they alive? Well, again, this seems to satisfy more elements of the checklist, but it's still somewhat unsatisfactory. Now, here we come to something that actually satisfies everything, and it seems to be very helpful to have that. Bacteria have complex internal structures. Bacteria have complex metabolism. They can maintain internal homeostasis. They can grow and reproduce. And some bacteria, for example, have these little flagella, these whip-like structures that allow them to tumble, and they can actually follow gradients. So they can avoid regions that temperature is wrong, or they can follow gradient towards a particular food that they need and ingest it. So they can respond to the environment to survive and find what they need in the environment. And when they reproduce, they do not make perfect copies. And again, that allows them over longer periods of time to change, which is, for example, why we are now dealing with antibiotic resistant bacteria in many hospitals. Because having found antibiotics that destroy bacteria, some of the bacteria, because they don't make perfect copies, are resistant to those antibiotics, and then those copies are the ones that survive. Okay, so this satisfies the checklist, but let's look at some other interesting cases. If you have a red blood cell after it's born, it loses its nucleus. But for quite a period of time, it's capable of still doing oxygen exchange, of making its way through your blood capillaries and doing its job. It hasn't fallen apart. It isn't. It's still doing its function. So, is it alive? It can't reproduce, but it's still doing its functions. Another example that people like to argue with is a mule alive? After all, it's sterile. It can't reproduce. Now, after a certain age, human beings often cannot or do not reproduce. I would be very uncomfortable saying that therefore they're not alive. So if you don't have something, an element on the checklist, does that mean you're not alive? So what this is suggesting is we need a broader way of thinking about living organisms. And so I'm going to return to that review I mentioned. And in reviewing the different theories of life that have been generated, they summarize a series of key ideas that they believe have to be incorporated into any theory of life. And we've mentioned some. I'm going to say a little bit more about them. So the first component is it should be thermodynamically open to allow supply of energy to be harnessed. This is actually fairly critical, as they made the point that living systems are capable of finding energy sources. And those energy sources may not initially be in the form that they immediately need. And so they have some way of assimilating those materials and transforming them into the things that they actually do need. And in part of the way they do this is by having these specific catalysts, these things that can actually transform one molecule into another. In some cases, what they transform are, in fact, themselves can act as catalysts. And it's important that those catalysts only primarily generate the appropriate set of things, because if they start to generate other things, these could be called parasitic reactions, and they could actually suck off some of the material that has to be going through a cycle and render it useless for the cell. we talked about catalytically closed because it has to be the case that all the catalysts that it needs are present and manufactured by it. Otherwise, it's constantly dependent on getting them from the environment. And then we mentioned this idea of being structurally closed. So most cells have a membrane that separate itself from other things that are not itself and from the environment. And this mentions also the importance of concentration gradients associated with these barriers that are used for energy management. A specific example that's very important for the things that I study is the gradient of ions across cell membranes that are used by neurons for signaling. The fact that there's energy used to maintain those gradients is actually used for particular purposes. And of course, things like pumping things into and out of cells require these concentration gradients be properly managed. It's very important that there's a mechanism for storing and reading information. The cells we're familiar with, that's generally DNA, and all of the associated machinery that's involved, molecular machinery that's involved in reading DNA and making it available to generate messenger RNA and then proteins. And then it should be capable of controlled growth. This is one of the points that Cornish, Bowden, and Cardenas emphasize very, very strongly, that if you don't have a way of controlling growth or regulating it, generally at the metabolic level, you have the possibility that many of the things that you generate will start to build up. This is very important. For example, the disease of phenylketonuria occurs because there's a particular enzyme that takes phenylalanine and transforms it into other molecules. And if that's absent, and this is something that a genetic defect that some people have at birth, then the phenylalanine starts building up and none of the subsequent things are made, and that can actually lead to retardation. It can be handled by someone having a very careful diet that is low in phenylalanine. But this gives you an example, a concrete example of how important it is to control the availability of substrate and how fast and how much, not just different components are manufactured, but also the growth of the cells that make up an organism. Okay. Now, what I also want to mention is that there are these major shapers of biological systems of cells and of organisms, and they are evolution, development, and plasticity. And I want to do a contrast between them and the way we deal with human-made devices. So in human-made devices, generally, if we're going to make a machine, we work through the design. And if we're going to program something, we often create an outline or a pseudocode version of that program. And that becomes a very important basis for what we do subsequently. One of the important things in human design is that if we make things, we try to make them as uniformly as possible, because we expect that those who are purchasing the things we manufacture are going to want them to all be the same. To give you a concrete example, if you opened up, you ordered from Amazon online a set of 6 glasses, and they came in and every single one was slightly tilted, had imperfections in the glass, discolorations, but they all held 8 ounces. You would probably send them back because you'd be very unhappy. Certainly wouldn't want to set at your table when you had guests with such glasses. But in contrast, if you went into a classroom, said hello, and 30 identical faces turned to greet you, you would be very, very disturbed. So clearly there's a difference between the way we manufacture things and what happens in biology. Finally, of course, it's not enough to simply make things. We have to be able to control them, whether they're complex machines like construction devices, or they may be various kinds of bots that are running in the background as we do our shopping online. All of these things are the way in which we do human-made devices, and the focus is on uniformity and on getting the same results every time if possible. This is radically different than what happens in cells and organisms. We'll say more about the details of this, but organisms are not designed. They evolve, and I'll say more about that. They develop, and that's how you make complex organisms. And they're subject to plasticity. This is very important for how networks of neurons change, but even individual cells are capable of forms of plasticity. And so now what I want to do for several minutes, and I hope you'll tolerate this, is to do a thought experiment with you. I want us to create a device, which I'm going to call, perhaps with a little bit too much grandiosity, a cell machine. And this will help illustrate the ideas in a concrete way. I don't think that right now this could be built, though I might be mistaken. And part of the reason of talking about this at a conference like this is that some people might be inspired by what I say and decide they want to try making it. That would be very interesting. So let's imagine you had a 3D printer that had some additional interesting properties. First of all, let's assume, and this is something that it is part of its specification, it has wheels and grippers so that it can move itself and it can also move other things that it encounters. It's programmable, and it includes a program that describes its own components and structure. It can wirelessly send and receive signals from other similar 3D printers. And finally, it is capable of making 2 copies of itself. And it's also capable of printing structures that are slightly larger than itself because of its mobility. Okay, so I've just given this artificial cell 4 properties. You can imagine we can make 3D printers quite small. Maybe it's a cubic centimeter on the side. Let's see what we could do with something like this. Okay, let's first point out that the importance of being able to make copies of yourself is really quite interesting. Let's say that in an hour it could make 2 copies, and that's all it does. It just makes those 2 copies and then it stops making copies. Well, in an hour, there'll be 3 devices, the original and 2 copies for a total of, so 1 plus 2 is 3. Now in 2 hours, the most recently made 2 copies will each make 2 new copies, so that'll be 4. So the total number of devices will be 1 plus 2 plus 4, or 7 devices, which I point out is 8 minus 1. And so, and you can show this by induction. If someone asks me, I'm happy to do it. You can show that in n hours, there will be 2 to the n plus 1 minus 1 devices. And that means that this is making an exponentially growing number of devices. So that's really quite interesting. Now, just making that many devices is, and again, if you just only could make, if the same printer only made 2 copies every hour, then it would make 2n copies, which is a far smaller number. So the ability to make copies of itself is already very powerful in terms of the numbers. But the other aspect of it that's very important is the motility and the ability to send messages. Because since they can send messages to one another, they can tell each device where to position itself relative to the other devices. So let's take a concrete example of what you could do. Each of the devices could say to another newly created device, line up to my left or line up to my right, and then you would end up with a set of devices in a line. Now you could go further because each device, because it's a printer, could print a surrounding shell which encased it entirely and was made of very stiff material. And that as that material dried, it would adhere to the adjacent devices. The consequence of this is that you could assemble a stiff structural element in parallel relatively quickly. You could think of it as a kind of a bone. Now, if other devices lined up, say above or below this stiff structural element and printed a soft bellows around themselves, and they had the ability to contract and expand those bellows and they attached to one another, then what you could have is the contractile actuator, which also could be assembled in parallel around the stiff structural elements. Now, the interesting consequence of this is, let's imagine you had 2 such stiff elements and you also had devices that printed a joint, like a ball and socket joint. And now you had actuators that connected at the top and at the bottom. And the resulting device, which had all been assembled in parallel relatively quickly because of the exponentially increasing number of elements that were moving into the right place, could move up or down. And so it could be used as a limb or a leg. Now, how would you use this device? Well, one way is you could try to hand design a controller for it. But another way is you could use something called genetic algorithms. So for those who are not familiar, just briefly review, I assume many of this audience have heard of genetic algorithms, but the basic idea is that you have a structure, let's say you have this limb-like structure, and you have some control signals to tell the actuators when to be active and when to be silent, let us say. And you can describe the parameters for that particular set of activations as a list of numbers. You know, time and amount of activation or whatnot. You could write down a whole string of those numbers. Now, what you do is you can start with random values, and you can create a population of strings that have the parameter values in them. And then you drop them either into the device or into a simulation of the device, and you see what it does. And you evaluate it based on whether it actually, for example, rhythmically moves the limb up or down, or even moves it at all. You could set up a sequence of fitnesses, just moving it at all, then moving it up, then moving it down, then moving it rhythmically. And if you did that, what will happen is some of the strings will have higher fitness than others. So in the next population, you take the best string and you might actually replicate it exactly, but the rest of the strings, what you do is, and including the original one, is you make small variations in their values. You could even take two strings, take the beginning of one and the end of the other and combine them that way. And so you have a new population where you've changed some of the parameters and you've built on the ones that are most effective. And the key thing is that the higher the fitness, the more replicates of the original string are present in the new population. So as a consequence, any variation that is successful will be overrepresented in the next generation. Now, if you do that over time, and this is work I did some years ago with Randy Beer, others have done this. We actually were able to show that if you had a locomotion, simple body for locomotion, you could use genetic algorithms, control the parameters for model small neural networks, and you could get this to walk at high speed. So this is clearly a solution that genetic algorithms can solve. Now, you may not want to start with a very long limb. You could start off with a much simpler device, perhaps just 3 of the printers making the structural, or 6 of the printers making the jointed structural element, and maybe 4 or 5 on top and bottom. But the cool thing about a device like this is that once you have it solved for a particular small number of elements, if you want a larger one, all you have to do is to have one more doubling period. And now you have twice as many elements, but since you already have rules for how they should be organized, it's not that hard to see how you could then use this to create much larger devices. Now, I deliberately just focused on one thing. There are many other things you could build with devices like this. If you think about it, only your imagination would limit what kinds of positioning, what kinds of things the cells, these artificial cells could put around themselves, and what kinds of very complex 3-dimensional structures they might be capable of making. I use this as a concrete example, but there are many other possibilities. Now, where did I get all these ideas from? Well, I stole them from biology, because all the processes I've just described are just a simplified explanation of how organisms are shaped by evolution and development. And I wanted to do it in such a way that you wouldn't be put off by using all sorts of foreign terms or seeing these strange looking fluid-like devices making copies of themselves, and you start thinking in terms of actual machines. But in the developmental process, you have a fertilized egg that divides repeatedly. Each cell has the DNA, which is a complete self-description of the organism, like the program that I described that has the self-description of the 3D printer. Cells have receptors for chemicals released by other cells and can release chemicals themselves and can follow chemical gradients. So that's where I got the idea of these wireless signals that the little devices transmit to one another. That's what effectively the cells are doing as a developing organism is being put together. And then I didn't go through this, but you can actually have some cells that die as part of the developmental process. So you would have a case where a printer would essentially eliminate itself, would secrete something that would take away all of its components, leaving a void where it had been. And depending on where the cell found itself, only part of the cell's description is accessible. So I had the example where one set of these formed the bone-like structures, so they excreted the materials that form bone, and others excreted the material that form bellows. Similarly, cells, based on where they find themselves, will do things like assume an identity where they create a very stiff matrix, hydroxyapatite matrix, and form bone, or they will make actin and myosin and form muscle, or they will elaborate long cylindrical structures and find tree-like excrescences from the cell body that allow them to take in lots of information and become a nerve cell. All of these are things that cells are capable of doing during the developmental process. And so what you have is exponentially increasing cells are positioned in polynomial time. And I want to emphasize that if we think about this, this is a remarkable thing. You have a fertilized egg in humans. Over the course of 9 months, you end up from a single fertilized egg to something that has literally trillions of cells in it and is capable of organized behavior. And that's seen in many other organisms. If you were trying to position each of those trillion cells, even if you had only a fraction of a second to do that positioning, that would take far, far longer to do. So this is a way of manufacturing that is very different than the way we currently manufacture devices. And one of the critical things is also that the process varies so that even identical twins differ at the cellular level. And of course, you have non-identical twins or who have just different siblings, they can differ quite significantly. Okay, and then when I spoke about evolutionary algorithms, I did so because I wanted to contrast the standard design approaches versus evolution. Here's a picture of the finches that Darwin described that he found on the Galapagos Island. He intuited, he didn't know this. Subsequently, Peter and Rosemary Grant did the studies to show that genetically this was actually correct, that these were originally colonized by one or just a few finches, maybe just a one mating pair that had come to the island. They were not originally native there, but they came over. There are various kinds of flotation rafts that animals can use to get to islands, or since they have wings, they can fly there. And what happened was in different regions of the islands, in some places, for example, where you had nuts that needed to be cracked, if you look at the top left, you see a very short, stiff beak. And in other places where there were, for example, cactuses, and you had to insert the beak into a very narrow area to get insects from the cactus, you ended up with a very thin, delicate beak. And all of these were initially from the original parents, but he saw that they were so different that they were classified as different species. This is one of the critical things that he recognized could be the basis for the origin of species. So what are the principles? Well, organisms vary. The variations allow an organism to survive more effectively in environment, and that surviving organisms leave more offspring. And variation is fundamental for evolution. It's actually the signal. It is not noise. So just as those strings of parameters I described have more, so to speak, descendants in the next generation if they have higher fitness, so organisms that are more fit in the environment leave more offspring and therefore come to dominate the population. And it's important to point out that during an animal's lifetime, process of selection acting on connections with the nervous system due to learning and plasticity allow for flexible changes in behavior. So there's a process of reprogramming that occurs in biological organisms during their lifetime because of the flexibility of the nervous system that they possess. Okay, now let's go back to our thought experiment and critique it just a little bit. So if we have devices that are subject to development and evolution, and I've shown you how the parallels, are they alive? And if not, what's missing? So I would not at this point claim that they're alive because first of all, can the device harness the supply of energy? If you go back to the criteria that I presented earlier that describe some of the key things, can they harness a supply of energy? I suppose they could print photovoltaic cells, at least some of them that were going to be on the surface, and others could have motility that moved you towards a light source. Certainly there are currently robots that can find, if they have an infrared beacon, they can find where the plug is and plug themselves in. So there might be ways of setting it up so that it could find energy, but that would need to be carefully thought through. And how does the device obtain the materials needed to create its components and distribute them to its constituent elements? Now, in biological systems, that's why you have, for example, a digestive system, which after you've ingested material, which is not in its original form, again, if you photosynthesize, then you directly turn photons into energy. So that's why I mentioned the idea of photovoltaic cells. But if you don't photosynthesize and you're dependent on eating plants or other animals, then what you need to do is you have to find them, you have to ingest them, and then you have to transform them. And that's what digestive systems are for. They're breaking down all of the components of what they've ingested into molecular components that can be absorbed and then can go to the appropriate cells in order to be further used for energy and for raw materials. And you have a circulatory system that allows you to ensure that cells deep within the body are never too far from sources of oxygen. That's what your capillary system is for. So that allows you to deliver the key gases that you need, oxygen, to make the ATP and to excrete the carbon dioxide that you no longer need because you've used it up in metabolism, and continue to persist. And your lungs and your heart are all critical for pumping, for getting that air in, exchanging it appropriately with that circulatory system, the elements, the red blood cells I mentioned, and then distributing that through the body. So there are ideas from biology one could think about, and one would have to do some hard work to try to design things that could make those kinds of elements internal to a three-dimensional device like this. And that might make it closer to something that we would call living, because it would be more autonomous. Once we made one of these, they might be able to manage on their own. But again, this would take some additional work and thought. And finally, how could you effectively control all of the elements to generate adaptive behavior? So I suggested one way using genetic algorithms, but what might make more sense, and is what biological systems do for very complex behaviors, they have specialized cells, nerve cells, that are organized in particular ways and are capable of complex and flexible control. So let's talk a little bit about that and see again what we can learn from biology about that. So what I want to talk a little bit more is about biologically inspired neural networks and say something about control versus plasticity. So classical control techniques, as everyone in this audience probably knows, can be used very effectively for linear systems. You characterize the input-output, the transfer function of the peripheral plant that you're trying to control. You determine what are the control variables that you have access to. You look at the signals that you are getting back from the plant. You compare them to a particular error, to a particular goal that you might have, whether that's a fixed location or a trajectory. You take the difference, that error can be fed into the system and be used for corrective action using the control variables that can control the system. And especially if you have a linear system, this works very effectively. And again, there's some issues in terms of dealing with feedback and delays and so on. It's not trivial, but it can. This is a well-established field, and one can do remarkable things with control theory. Now what's happening to deal with nonlinear systems is people are starting to use artificial neural networks, and they're starting to show significant efficacy. So that's really very exciting. And it's of course worth pointing out that these are inspired by biological neural networks. However, they have some significant drawbacks. They do require very large training sets. Once you get one that works, you just have a set of connections, and it's really hard to understand what those connections mean or imply or how they work. And these can be, in some ways, quite brittle. People have shown that you can, for example, on the sensory end, you can get networks that are well tuned to particular sorts of things. You make a small change, they no longer recognize those objects. And that small change is something that would not bother a biological neural network or visual cortex. So something is missing, and again, it's very hard to figure out what the differences are and why they are that way. In contrast, it's important to recognize that even animals that have relatively small numbers of neurons are capable of rapidly learning from experience and can flexibly alter their behavior. So we have 10 billion neurons, that's a lot, and trillions of connections, hundreds of trillions of connections between them. But even an insect, which has about 1 million cells, can do remarkable feats and can figure things out. And the sea slug that I love and study has about 20,000 neurons, and it's capable of feeding and locomotion and mating, and it can learn, for example, that food is inedible. And animals, from the time that they come out of whatever enclosure they're in during the after development, whether it's an egg or they're born from a mother, are usually able to do significant numbers of things on their own. Once a Aplysia hatches, it's a free-swimming veliger. It has these velar lobes, and it can be a filter feeder. And then once it transforms on Laurencia, the seaweed it loves, it becomes a little slug, and it immediately knows how to feed on seaweed. Doesn't have to have any training. It can learn, however, which parts of the seaweed are inedible and which are edible, which are very important for its survival. So the question this raises are, what are the features of biological nervous systems that allow them to be so effective? And that's what I'd like to spend a little time talking about now. Okay, so these features are well known to neurobiologists. Many people who have an engineering background, and the last time they confronted biology was perhaps dissecting a frog in high school. And after looking at a textbook filled with lots of weird names and complicated structures, all of which we're supposed to memorize, they closed the textbook and said, "I'll stick with math, thank you." And I can actually appreciate that viewpoint. But I'm going to just give you a high-level description of what it is that makes real neurons so effective, and perhaps reinterest you in learning more about the biology. Starting with Pitts and McCulloch, the idea was that neurons were just on-off devices, and they basically could summate the inputs, and they could more or less then make a decision whether to be active or not active. And in fact, in artificial neural networks, you've got the various weights coming into a neuron, and a neuron is represented. It's a little bit more sophisticated than just 0 or 1. It's a sigmoid function, which would represent a continuous range from being off to being on. But that's actually a very, very simplified caricature of what neurons actually are. Neurons are actually each complex dynamical systems. What do I mean by that? An individual neuron has in its membrane a series of channels that allow certain ions to come through, and these channels can be gated. They can be opened or closed by either the voltage across the membrane or chemicals that bind to the membrane. The consequence of this is that neurons can actually generate activity in the absence of any other inputs. They can be spontaneously active. They can generate rhythmic bursts, and those rhythmic bursts can be very complex. They can fire fast initially and slow down, or they can fire slowly initially and speed up. And they can fire irregularly, and they can be silent. And even more importantly, they can have multi-stability. That is, as a function of the inputs they're getting, other chemicals around them, they can shift from being completely silent, for example, to rhythmically bursting and back. So this is the characteristic of what we call a dynamical system. It's something that shows complex processes over time. And it's certainly not just an on-off device. Now, that complexity is further enhanced by the shapes of neurons. Neurons have a wide variety of incredibly remarkable shapes, very complex shapes. Ramon E. Cajal, working in his kitchen, this is before the era of NIH grants, using the Golgi stain, would impregnate these blocks of tissue. And then he was a very good artist, and he would draw these phenomenal pictures, which, you know, if you hadn't subsequently seen this, you would think he was just hallucinating, because they have all these elaborate structures. Neurons have very different shapes. Now, the reason that's important is this: where an input comes in. So let's say this is the central part, the cell body of a neuron, and the decision point as to whether it's going to start to fire. Let's say it's down here. So if an input comes into one of these little fine branches called dendrites, if it's out here, it has to travel electrically down to here to activate. But if it comes in much closer, like here, a much weaker input can generate the inputs. So a whole series of very strong inputs can come at the edges at fast frequency and turn it on, or a few well-placed inputs can turn it on. It's the difference between, for example, voters in a democracy trying to set the direction of their country versus someone who has the ear of the executive and can immediately get something done because they can just talk to them and get them to approve something. So a more general way of saying it is that neurons respond to complex spatio-temporal patterns of input. So that's rather different than just summing up weights. Okay, and the neural connections themselves, the connections between neurons, have complex dynamics. And depending on how they work, they can be private. The chemical that's released can just talk to one other neuron in the synapse within the synaptic cleft, and that's like a private telephone conversation. Or like a peptide can be released and can affect not just the adjacent neurons, but some neurons in the vicinity over a longer time scale and a longer spatial scale. That's like somebody having an active conversation on the bus, something I always try to ignore, but I can't so easily do because they want me to know about what they're doing for dinner and what so-and-so said to them yesterday and so on. They feel the need to share. In the nervous system, this is actually rather useful, whereas on the bus, if you're trying to read and think, it's somewhat annoying, but unless you're really interested in overhearing other people's conversations. And then finally, they can act at very long groups, and large groups, and very long distances. They are almost like a radio transmitter, where if you're tuned to the right channel, you can get the message, you can get the signal, you can hear the music you're interested in or the news from great distances. You just have to have the receiver tuned properly. And that's like hormones, which as long as there's a receptor on a cell that can bind it, the cell can respond to that message and act on it. That's very different than the kind of uniform connectivity pattern that you see in artificial neural networks. Another key thing is that what the artificial neural networks are doing is they're trying to adjust the specific connections as a function of experience. But one of the issues in nervous systems is there are ways of tagging which of the connections are important as a function of the recent experience, and those are the ones that get changed. Not all the connections, but the relevant connections. In some studies I've done with my colleagues in the slug, when we look at the connections that are changed after you teach an animal that food is inedible, those connections that are important for rejection behavior are actually strengthened between the sensory and motor neurons, and the ones that aren't are not. So you can have selective changes in plasticity. Another important issue is, again, the architecture now for artificial neural networks is you have at least 3 layers and maybe more, but they're just layers. And then from each layer, there's a huge number of connections to the next layer. That's not how neurons actually are set up. There's a complex architecture, and that architecture gives rise to things like cortical columns or the cerebellar architecture that's able to transduce information in very complex ways. And so architecture matters. It's very important. And another important issue is that the control can be shared with the periphery and the environment. So for example, if you watch an eagle, at some point as it's looking for prey, it may be riding a thermal. It's not flapping its wings. It's not doing anything with its body at all except riding that thermal. So its nervous system is being used to look for prey, but it's actually not flapping its wings at all. It's using the environment for flying. And then of course, if it sees something interesting, it starts to flap, it drops rapidly, and goodbye to whatever it is that it's cited. Similarly, if you have a heavy leg, this is one of the points that Greg made in his excellent presentation yesterday, then at the onset of swing, if you have a heavy leg, the impulse to the leg is sufficient through the inertia of the leg to keep that swing happening. And in fact, you need another impulse on the opposite set of muscles to brake and slow that, because inertia otherwise would keep you going too far. So you're sharing the control with the periphery and with the environment. And control emerges, and this is the key point I want to make and leave you with, that control in the nervous system emerges by shaping neural dynamics, peripheral environmental dynamics, whose initial properties are set by evolution and development. And one of the ways that we are trying to take advantage of this, you'll see presentations about this, is by creating what we call synthetic nervous systems. This is work that Nick Shazinski and Roger Quinn have pioneered, and there's several presentations in this meeting, which I encourage you to listen to, where the neurons have some internal dynamics, they have interesting architectures. They're still simpler than real biological neurons, but they're a step closer to the way real neurons work. And therefore, I think that's one of the reasons why one can do some very interesting control with them that one can't do otherwise. Let me briefly talk about directions, the other direction. This is all talking about how one can try to create artificial devices or simulated devices that are biological alike. But what's going on right now is in the biology side is absolutely astonishing. Synthetic biology now can create designer genes cells, organs, and even organisms. That's a realistic possibility. There was a really nice article in the New York Times Science section a few weeks ago about the new uses of CRISPR-Cas. CRISPR-Cas allows you to snip out pieces of DNA and insert other pieces of DNA. So if you want tomatoes to make a particular kind of compound they never make naturally, you can get them to do that for you. This very controversially, someone actually tried to do this in human embryos, a Chinese investigator who now I think is in prison, to try to create HIV resistance. But the possibility of changing these things and engineering them is now becoming possible. And it's very interesting because if you work on the germline, things that affect the manufacture of sperm or eggs, then it's a hereditable change. It's not just a change that will be in the lifetime of that organism and then die out with that organism. All of its descendants will have that change. And another direction that people are going is to try to simplify down things like mycoplasmas that are very, some of the simplest organisms, and see, can I eliminate many, many different elements and create a minimal cell that could then serve as the basis for creating novel artificial cells. And people are also working very hard at making excellent progress on developing simulations of these minimal cells, which allows you to begin to explore how they'll behave in response to both internal and external changes. Now, before I end, I just want to point out that all of these things I've been talking about have very important ramifications that go beyond the excitement of just let's make something cool and new. As we know, for example, the Great Lakes were recently, some years ago, the quagga mussel was introduced probably from the ballast of ships that use the Great Lakes. And these quagga mussels secrete this gooey stuff and they outcompete all the natural mussels and they clog the waterways. And they're an invasive species. They weren't there originally. Now, as things are warming up, species that would tend to die out through the winter and then couldn't go beyond certain regions are now beginning to evade new regions. And this is all happening naturally. But as we see, when that happens, that can have massive disruptive effects. We've just been experiencing and are still experiencing how a pandemic, a virus, which was not, I believe, released by humans, it was probably jumped from an animal to a human is the most likely explanation based on the evidence. But they show how new variants of bacteria can have major impacts on human populations. So the problem here is that if we succeed in creating artificial self-replicating devices that can modify themselves in response to environmental conditions, they could have profound social, economic, and political impacts. At the same time, therefore, that we work to develop these devices, I think it's very important to make sure that part of our teams involve ethicists, and we must encourage regulation to ensure that these devices are properly controlled. It's a little bit too late when the thing has escaped from the laboratory and is now devastating the population. You go, oops, I didn't mean that to happen. We need to do some work in advance of that and get ahead of it. And then if we make this part of our concerns in the years to come, then I do want to end on a very positive note, because some of the things I'm discussing, and I hope some of you actually got excited by this cell machine that I described and its potentialities, we have the possibility of reaping remarkable benefits. And we could avoid the considerable harms they might otherwise cause. So I'd now like to wrap up with a summary and conclusion. The first point is that cells and organisms can be thought as complex molecular machines. That's a term that you'll find people using. So that answers one of the questions that I raised. Are living things machines? People call them molecular machines, but they're a very special type. They constitute a network of processes that produce the key components necessary to maintain the operation of the network as a unit. They're able to assimilate energy, all the other things we talked about. But the key thing is that once you set them in motion, they can, so to speak, stay alive. They can take care of themselves. You don't have to plug them in. You don't have to change their batteries. You don't have to make sure that they're positioned in such a way that they're receiving the appropriate signals. They just do all that stuff on their own. There's a fundamental autonomy and self-containedness that seems to be critical for these kinds of molecular machines. I pointed out, I hope this, by making it very concrete and painting these pictures for you verbally, that concepts from evolution and development could be used to create artificial devices that could very rapidly self-assemble and could be updated through creating and evaluating many variants, either of the actual devices or through simulation. And I talked about how synthetic nervous systems, by incorporating many more features of biological nervous systems, may make it possible to control complex behaviors and adjust them effectively over time. I also pointed out that new advances in synthetic biology and what's called artificial life may soon create devices that really almost completely blur the border between the living and the non-living. And I concluded by emphasizing that the major impacts such devices can have socially and politically require thoughtful consideration of ethical concerns and careful regulation. Now I'd just like to thank several people. Many of the ideas that I'm talking about, especially focus on autopoiesis and genetic algorithms, were done with my colleague and friend, Randy Beer. Roger, Quinn, and I have been working for many years to create biologically inspired robots. And I don't know whether he's now going to create a new branch of his lab to create cell machines. I wouldn't put it past him. I've also had the privilege of working with Victoria Webster Wood, who you'll be hearing from on Friday, on developing biohybrid robots, and with Greg Sutton developing concepts of understanding how biomechanics affects energy distribution and also neural control. I've had a long-standing collaboration with Peter Thomas, who is an applied mathematician, and we have used some of the things I talked about, some of the simpler models of these neuromechanical systems to understand more about robustness, flexibility, and how to deal with real-world conditions like changing loads. My colleague Abraham Sussman I mentioned briefly because I talked about the learning work that I've done with him. And the original model of the neuromechanics was done with Kendrick Shaw and was developed further by a postdoc that I shared with Peter Thomas, David Lytle. A vital member of my laboratory for many years has been Jeff Gill. Much of the data we've gotten is from him, but Jeff, in fact, started with an interest in autopoiesis, and I hope that he continues that interest. Some of the students who got the data that I mentioned for the learning project, which will meet Tom and Itai Horowitz. Another critical person in my lab is Jade Yuan, a graduate student who's been getting very important data for our understanding of the dynamics of the neural network we study. Yanjun Lee will be giving a talk. He is co-advised by Vicki Webster Wood, Greg Sutton, Jeff, Roger, and myself on a artificial neural network, a synthetic nervous system for controlling feeding aplasia and showing that it can be multifunctional. I'm very grateful both to NSF and NIH for their funding of my work. And with that, I'm happy to take any questions that you may have. Thank you for your attention.
Thank you very much, Hillel. That was very, thought-inspiring. It's I always have questions, but we'll leave it up to the audience.
Right. Well, I think Jeff has summarized it by saying, "I, for one, welcome our new living 3D printer cell machine overlords." Thank you.
Questions? Paul.
Yes, Paul.
I knew Paul would have questions.
Yeah, look, okay, this is really great thought experiment, right? And great analysis. But the theory of evolution is also complexifying with the day, right? So now we are in sort of the extended modern synthesis, where also new layers of control loops are brought in, right? You have evo-devo, right, where you also start to have now environmental impact on transcription, but also beyond that, niche construction, parent selection, cultural evolution, which are now all brought in as additional levels of organization, feeding back in the end into this whole notion of the change that might be due to heredity. So in the proposal you're making, Hillel, how would you then include these additional levels of control loops? Of course, we can say, oh, it's accounted for, we can do it, right? But that also would require another level of engineering that would go beyond 3D printing the physical instantiation of the exemplar, right? Of the phenotype. So how are you going to solve these additional layers? Because otherwise I cannot get started.
Well, first of all, I think it's important to recognize that even though these various aspects are important, there are simple organisms that are capable of perfectly well replicating themselves, like bacteria, that don't seem to use the higher levels at all. And they're nevertheless capable of doing some very useful things. So you don't have to have these things necessarily to get started. That's what evolution shows us. The second point, though, I take very much what you're saying. The issue is that if you talk about these self-organizing elements, like these cell machines, I commented very briefly that the lineage and the position determines what you do next, what part of your self-description is activated. That's what happens in development. So if you wanted to incorporate into that certain epigenetic factors, such that certain parts of the self-description are unreachable as a consequence of something that has happened in the environment, or they are activated because of something that's happened in the environment, I don't think that's beyond the reach of the idea that I suggest. Now, if you have these we'll call them artificial multicellular devices interacting, and they actually have synthetic nervous systems, the possibility of fairly complex behavior emerging from within those groups is certainly possible. Beyond that, I don't really want to speculate, but I just say that the history of evolution is such that you can get increasing layers of complexity starting from great simplicity. That's what the fossil record seems to suggest. And therefore, I don't think that it's beyond the reach. But I agree with you that there are many steps that need to be made, even to implement what, and this is why I was self-critical, even to implement what I suggested as a thought experiment, dealing with the question of getting the energy, of distributing the energy and distributing the raw materials is non-trivial, would take a great deal of work. But if we start with something simple, the beauty of both evolutionary and developmental processes, as I tried to point out, is that you are not starting from scratch. You can build from good initial conditions and make things even better. And that, I think, is really the profound message.
But now in this Cornish-Bowden review that you started out with, which actually is a very useful paper.
Yes.
You come out with a checklist, right? But also actually they do emphasize this core concept of autopoiesis, of sort of self-maintenance, quite a bit in that article. So I also could imagine you would have taken that as your core feature, because that sets another set of criteria. So why did you move away from that and went for a fuller list?
The reason I did that was because one of the things that was so nice about that particular article is they talked in detail about the hypercycle approach, the MR systems, the autocatalytic systems, and the chematon. And they pointed out some of the defects that are found in each of the different models. And what I was pointing out was not so much a list as a set of desiderata for a theory, whatever that theory might end up being, that one would want to ultimately develop. So I actually have been very influenced by the concepts of autopoiesis. My hesitation with it has been, and this is why I found that article so very helpful, is that in terms of actually creating simulations, I mean, Varela tried to do that in the 70s and was able to actually get something to work again in the 90s, but it was fairly primitive. And one of the things that those reviewers are actually have done, they have created very detailed and much more realistic simulations that have some of those properties. And they've been inspired not just by autopoiesis, but by the other ideas. So I wanted to give credence to that larger group of ideas rather than just focusing on one.
Okay. Great. No, thank you very much. This was a great way to get started. So thank you very much.
Thank you for your kind words. I appreciate that.
And Nick, you had a question. You seem to have dropped your hand though.
Yeah, in the interest of keeping everything on time, I'll catch up with Hillel later. But it was a great talk, Hillel. Great scope, great way to get us started, like Paul said. So thank you.
I appreciate that. And again, if someone is inspired by what I've said to actually start trying to do this, I'm sharing these ideas. And I freely emphasize that they were inspired by the biological system, which is why I'm giving them away. But if you found it useful to talk to me about them, if you tried to do this, people have worked with me in the past on biologically inspired robotics and other things like that have found it useful to get some of my insights, and I would be happy to be involved in those discussions. So I guess I could leave it at that.
Okay, but let's thank Hillel again for a beautiful presentation. It was great. So we have a break until 20 minutes after the hour. And we'll see you again in about 17 minutes.
See you all. Hey, Vicky.
Good morning. Hello.
Good morning. How are you?
Doing good.
I see a whole
Yeah.
group. I see a whole group there.
Yeah, we got set up for a watch party. A bunch of our friends drove up from WVU and over from Penn State so that we could make it feel a little less virtual.
Uh-huh. Very nice. Well, I hope I didn't put you all to sleep.
Oh no, it was an excellent plenary. Very thought inducing.
That was what I was hoping I would succeed in doing. And I thought this audience would probably appreciate it more than a standard biology audience would. So I felt a little liberated to do things I wouldn't dream of doing if I were giving a neurosciences talk.
Yeah, it's a good audience for it.
Yeah, yeah. Well, I can tell you that you will be a much happier person an hour after the meeting starts on Friday. That I can definitely tell you.
Yep. Yeah, I'm still polishing a little, taking notes as I watch the other plenaries.
Yeah, I was up late and up early and continue to be thinking about it even as I was giving it. So.
Yep, I'm sure I will do the same.
Anyhow, if in your talk you then propose that you're going to start building the first cell machines, I'll be very excited.
We do a little bit to a certain extent. Not quite to the full extent of the thought experiment, but hopefully one day we'll get closer.
Yeah, yeah. So is it really hot there in Pittsburgh as well?
It is fairly toasty here, but it was pretty nice this morning.
Yeah. Yeah, it's going to be getting into the 90s here.
Yeah, I think we're supposed to be pretty warm today too.
Yeah.
Well, I actually had a quick question. If you,
Great.
I was, the talk was awesome. It was really, it really got me thinking about a lot of things. And one thing that actually came to mind was I was kind of wondering what your personal thoughts were on our current abilities in terms of technology to control evolution. And like, are we taking it too far by manipulating and taking things into our own hands in some regards? I just wondered if you could speak to that a little bit.
Yeah, so it's a very interesting question. So it happens, ironically, that this is a question that my son, Josh, who is in physics, has been working with Mike Hinchevsky at CASE on, and they've actually been using concepts from, it's called counter-diabatic driving theory that's used in quantum mechanics. They've been using that on simulated systems to show that they can actually, to some extent, drive the evolutionary process. So for example, if you have a drug that's going to be used against, say, a bacterium or cancer, and it's going to tend to develop resistance, that by setting things up properly, you may be able to actually drive it into a condition where once you use the third drug, you'll actually wipe things out. You're asking a broader question, though, which is as soon as we do things like, for example, have people wear glasses or save organisms, save children that would have ordinarily died young, or apply various kinds of antibiotics to wounds that were often fatal, we are changing a great deal about the human population, and that is going to affect our evolution. And we're doing an uncontrolled experiment to see what happens. I'm at this point somewhat more concerned about what we're doing to the climate and the fact that our cerebral cortices, because we tend to focus on really short-term things, seem to be incapable of grasping the fact that if we take action earlier, we would have less problems later. That is a source of frustration to me. But we are absolutely doing things that change evolution in very significant ways. That's not the same as controlling it. That's modifying it in putting it into different directions that it might not have followed otherwise. The more serious issue, and I was trying to raise this very briefly, is that if you can edit genomes, then if you know a child is going to have a defect, you might be able to edit that out. But you could do more, and you could, for example, give the child blue eyes and brown hair by appropriate editing. Are we going to go that route? That could radically affect the sex of the child. Anyhow, I think we're ready for the next talk, so I should probably stop. But there are some very deep issues that are going to need to be discussed because of the things we can now do biologically.
Thanks. Yeah.
So yeah, that was a nice discussion. I say hello to everyone from Italy. Here it's also very warm, and yeah, very welcome to the next session, which will be on bio-inspired robotics. And the first talk will be given by Falk Tauber, who is working at the University of Freiburg. And he's working in the Cluster of Excellence for Living, Adaptive, and Energy Autonomous Materials. And yeah, his talk will be about a unit cell-based artificial Venus flytraps. So Falk, you can start the presentation.
Yes, thank you very much. Yes, thank you, Kenneth. We are actually going into 105 degrees Fahrenheit right now in Germany. So yeah, we're also a bit toasty, I would say. So thank you very much for the nice introduction, Fabian. As he already told, I'm working at the LiveMats Cluster of Excellence in Freiburg. We are actually trying to develop something like a cell machine, as Hillel already showed you. Yeah, we're trying to develop living, adaptive, and energy autonomous material systems. And one of our goals is to develop a artificial Venus flytrap that actually uses these systems to catch prey and energy harvest autonomously. So I'm going to tell you something about our newest addition to the series of artificial Venus flytraps that we're developing, and this is our unit cell-based version that you see here. And the key question that we want to answer in our project is how to build an artificial system that is actually indistinguishable from a living system, or to be more precise, in our case here, how to build an artificial Venus flytrap that would pass a Venus flytrap functional Turing test. What do I mean with that? So if you would, if an alien would come to our planet and would see our artificial Venus flytrap and our biological model here, and he would say, okay, it looks like the Venus flytrap, it moves like a Venus flytrap, so it should be a Venus flytrap based on the functions. It would not be able to discern which is which, so one is fake, one is real, but he wouldn't be able to see. And to answer this, we need to know the key characteristics about the Venus flytraps. And here you can see we have a Venus flytrap here. It consists of two lobes connected via a midrib. It is able to discern two distinct stimuli and react with a closure movement after this two stimuli within 20 seconds with a release of stored elastic energy and in an elastic deformation of these trap lobes, and close fastly, so it's in a snap buckling motion, these trap lobes here within 100 milliseconds to 500 milliseconds. So key characteristics would be the fast snapping motion, the sensing, a decision-making capability, because it should only close its lobes when there's prey, not a raindrop that only hits a trigger hair once. And it should be able to harvest energy not only from the prey, but also from its environment. And there are already many different Venus flytrap, artificial Venus flytrap systems out there, but they're all mostly byproducts of interesting material development or a development of an actuator system. So there we have systems that are driven by magnetic fields using magnets with these attached to these pre-band structures here. We have systems that use Joule heating via shaped memory alloy springs to close these structures for the closure movement. We have systems that use electric fields that drive ionic polymeric metal composites and dielectric elastomers, or humidity-driven systems that react to a change in environmental humidity using hygroscopic bistable sheets or hydrogel swelling to move the system and close it. We have pneumatically driven systems that actually close or trigger a bistable system here to close these concave leaves. And we have photothermally driven systems that are based on liquid crystalline elastomers or liquid crystalline networks. But not all of them, not all of these artificial Venus traps integrate all Venus flytrap functions. Not all have sensors. None of them have actually these energy harvesting capabilities. None have the capability to do decision making, and none incorporate more than one of these motion principles. So mostly the closing, but not on reopening. And we actually thought about abstracting these trap lobes into a metamaterial, because metamaterials are able to we can incorporate this bit of decision making and the sensing capability already at the moment. And we want to also integrate energy harvesting in such systems. And we want to use our living and artificial living material systems that we develop in our LiveMats cluster to bring these artificial Venus flytraps to the next level. So how can we actually transfer this motion that we saw earlier into a metamaterial system to create this adaptive system here? We use an auxetic inspired mechanical metamaterial, shortly AIM, as an actuator for our artificial Venus flytrap. And this is based on the actuator, the system of the kinetics from Wu et al. From the MIT. They developed a basic bending, basic unit here that is based on a polygon type auxetic material simplified at this 4-bar linkage, and it is free in the position of the joints, joint angle and wall angle, and through that they can generate planar transformation or spatial transformation like bending or twisting here. And we want to combine these possibilities here to create this doubly curved surface that we see from the Venus flytrap lobes. And we use for that PolyJet 3D printing system, because we can there combine flexible and rigid materials in one setup. And we used for a first test, developed bending strips and made a combination of a basic and bending unit here to see if we're actually able to get this bending and curvature that we wanted to see. And if you compress one of those outer units or a central unit, then this leads to a 2D bending that you can see here. But the transformation of this one unit cell was not well forwarded. So we had to see, okay, that our curvature is not strong enough for our purposes. So we had to see how was the curvature. We set points along this model, approximated the curvature here, and computed the curvature, and saw that the curvature was actually not really consistent in this model. So we changed our bending strips to a model with completely only bending units. So no basic units are in here. And we changed the height of the units. So you can see here the first bending unit with the basic and the bending units here. The bending strip with 3 millimeters, and here we have without the bending units, without the basic units, and a height of 7 millimeters. And we had added also a hardness gradient so that we could achieve higher curvature. So when I say hardness gradient, I mean within these flexible joints here. So this is the hardest, and then the shore hardness goes down from further away from this central unit here. And what we could see was that we achieved a lower curvature with these 3 millimeter high bending units the small unit that you can see here gets already a bigger curvature with the 7 millimeter height. And when we have the longer strips, we could actually achieve the highest curvature. But we also found that the hardness gradient actually had not only marginal effect on this. So we had our first bending strips with good curvature. So we now wanted to develop a double bending strips to see if we could actually activate them with a central unit here. So we added here a basic unit and changed also there, or investigated if the change of the system and the height and geometry of the central unit would affect the curvature. And what we saw is that we actually got a better curvature and higher curvature with a bigger central unit, and that the hardest gradient also had no significant effect here on the curvature. So what we did then was to see that we already had our 2D bending strips and that there was no torsion, so we wouldn't get this doubly curved surface. So to achieve this 3D curvature that you can see here, like in our Venus flytrap here, we created a new design to achieve this doubly curved surface with symmetrically tilted opposite walls here. And we achieved with that our first doubly curved surface. And we used the flexible hinges to test our designs here, because they're really easy to print, but they showed a poor durability. So after around 1,000 cycles of actuation, they would deteriorate. So we had to change the hinges here, and we choose to change them from these flexible to actual barrel hinges. And with that, we actually achieved a higher shape change, so we could generate even more higher bending angles. We added, unfortunately, a bit of joint fraction into the system. The miniaturization is actually the same as with the flexible materials. It has a bit of a higher design effort, and the variation of the joints isn't as high as the flexible joints here, but we added a extremely higher durability. So these, the first ones that we built, broke after 15,000 activation cycles, and then the biggest, the newest one, they didn't break yet after 50,000 cycles. So with that, we then created our first doubly curved surface system completely. We added here the central unit. We have our bending strip and added with the doubly curved surface, and we were able to fully 3D print this in a full assembled state. So the only thing that we needed to do after it was printed, we needed to clean those small hinges here, but not assemble each of these unit cells. This was a quite good achievement here. And what we then were able to test was how fast we actually can go with these models. And we saw that we actually were able to achieve this doubly curved surface inversion here. And on average, it took 250 milliseconds for closure, but this is highly depending on the applied force here. And we attached a foil as a carrier for sensors and energy harvesting structures for the future development of the system. And we saw that the foil did not really affect the closure. And we tested this also in the closure speed here. And that you can see the velocity, the tip velocity is not really impacted by the foil. Actually, it got a little bit faster. So with this system, we actually were able to design a first artificial Venus flytrap that is even faster in velocity terms as the biological role model, and snaps or closes within the same time range, and is able to show this curvature inversion of the trap lobes here. And it already also looks like in the Venus flytrap a bit. But we want to see now how much is actually the force needed to activate the system, because we want to now then incorporate in the next step of the project here environmentally driven shape memory polymers as a central actuator. And what you could see is that the curvature inversion, so passing this 0-degree curvature, takes roughly between 0.8 and 1.3 newtons. And we tested this with 3 different demonstrators, each 25 times, and you see that there's only a marginal difference here. And we were able to show that we have actually this dip in necessary force after the 0-degree curvature, but that the full closure takes roughly 2.4 newtons due to the friction in the hinges and the hysterical hindrance of the whole system. But what we also were able to show is that with lubrication of all of these hinges, with a silicon-based lubricant, we were able to put down the amount of force needed to 0.3 to 0.5 newtons. So we're here really in the range of force that low energy shape memory polymers could achieve. As you can see here also is the 0 degree curvature point. We see this dip here. We investigated this with a parameter response measurement here, as you can see at these orange parameter here. And when this is represented here in this graph with this orange line, and when this, just a second, going through the 0 degree curvature point, so when it's all is the biggest parameter here, then it's the 0 degree curvature point. And you see here that here we have really the drop in this necessary force. And we will do now additional tests in horizontal orientation to investigate possible influence of gravitational pull and incorporate our shape memory polymer actuators in the central unit here. But this first characterization of the system showed a successful development of our mechanical metamaterial unit cell based artificial Venus flytrap. And now I would like to shortly summarize my talk again. So just to show you the key question that we were able to show a little bit how we could build such an artificial system already, because our artificial Venus flytrap already mimics the appearance and the speed of the Venus flytrap and its snap buckling motion. It actually outperforms the model in the movement speed and passes our Turing test for the Venus flytrap in the motion part. And we want to integrate these shape memory polymer driven actuators in the central unit. And they are experimenting with bistable systems here. And in the future, we will add the sensing and energy harvesting structures onto our foil system and attach this to the inner parts. And the motion pattern and the system could be used as the backbone for energy harvesting structures for autonomous soft robots and novel gripper technologies. And with that, I'm at the end of my talk. I would like to acknowledge all our our colleagues and our funding and the Living Machines Committee to choosing my talk for here. Thank you very much.
Thank you very much, Falk, for this very nice presentation. We have time for 1 or 2 questions. Please, if you want to ask a question, just write it in the chat, or better, just unmute yourself and ask it directly.
All right, Falk, I have a question.
Yeah, sure.
Really great talk. I love this metamaterial thing. This is super cool to me. However, I can't help but notice that this prototype seems to be much larger and much more massive than the flytrap. Is this mainly a study of how to get the kinematics and how to assemble the metamaterial?
Yes, at the moment we had this first study to see the kinematics and how to assemble it, and we are currently printing this in our Up Nano or NanoScribe. I don't know if you guys know these systems. These are 2 photon lithography 3D printers, and they have up to sub-nanometer resolution and a build height of 1 centimeter. So we're currently, and it takes roughly around 1.5 days to print one of those systems. And unfortunately, currently we don't have a functioning one, but we were able to print these barrel hinges and a small scale one already. But yeah, we're focusing here in this study on the kinematics, and now we're currently miniaturizing the whole system.
That's fantastic. Wonderful. Thank you very much.
You're welcome.
Okay, I see that there's still 2 hands raised. Please go ahead, Vicky, maybe.
Yeah, this is Saul. I'm one of Vicky's PhD students.
Hello.
And I had a question about the nature of the deterioration of when you were originally using the rigid and the flexible materials on your polyjet. Sure. Can you talk a little bit more about how that failure occurred?
Oh yeah, so I don't know if you're you guys know these PolyJet printers, but they are actually, you can think about that they're putting layer on layer on layer, and then they are UV cured. So it's an inkjet system, and we were able to get a better connection from these parts, so they're printed directly connected by each other if we used a mixture of both materials. So they mix the rigid and the flexible material to create these hinges. And if we have the mixture, then it is a bit better adhesion of the whole hinge to the rigid part. But if we wouldn't have a mixture of these two systems, then it would just peel off after 1000 cycles. As a full flexible hinge would just start to peel off. And we had an issue with hardening after a few thousand cycles, that these hinges started to harden further due to UV light, or just due to the material itself. And they also just started to tear. So that was the main thing. The tearing was the biggest problem. We tried to use hourglass shaped hinges then, but we couldn't get the tearing away. So we just had to say, okay, this will not suffice. And we changed this to these barrel hinges, and these are actually really good. So they really hold, but they're tedious to clean actually. So it takes roughly 1.5 hours to clean one of those 3D printed versions.
Wow. Well, thank you.
Yeah, you're welcome.
William, if it's a very quick question, you can go ahead.
It is.
Quick answer.
It's very quick. Great talk, Falk.
Thank you.
My one question, it's a silly question, probably. I noticed that the quote unquote real fly trap and your older artificial fly trap both start almost closed, like the curvature is in the correct orientation. And the new one that you're doing is working a lot harder because it's starting either completely horizontal or even curved the opposite direction.
Yeah, we wanted to
make it harder for yourself.
Yeah, that's a good question. Thank you. That's a good point. We wanted to actually create a big surface, so because we want to attach our flexible solar harvester systems on the inner surface of these. So if we want to then have a rainstorm or dust storm or something, that we can close them up and keep them in a safe bubble, for example. That's why we created these big ones. Yeah, sure. This one here, as you see, it already starts halfway closed. And that's the, and we wanted to just see that we can actually also have the harvesters and expose them to the most part of the light here.
Gotcha, that makes sense. Thank you very much.
You're welcome.
Thank you, Falk, again. Let's move to the next talk. So the next talk will be by Atsushi Kaneko from the Umedaki lab at the Shinshu University in Japan. And he will talk about energy conversion of elastic energy stored in the legs of a hexapod robot into propulsive forces. Looking forward for that. Please start your presentation.
Hello, my name is Atsushi Kaneko from Shinshu University. I will start my presentation entitled Conversion of Elastic Energy Stored in the Legs of a Hexapod Robot into Propulsive Force. This study proposes a novel leg design method to increase the speed of legged robots using flexible materials. The method can design flexible legs that convert the elastic energy stored under ground reaction force into propulsive force. The experimental results show that the legs designed by this method can increase the speed of up to 89% compared with existing methods. Let me explain the background of this study. Inspired by animals, many legged robots with flexible and elastic legs have been designed to achieve high locomotion performance. Animals can use the elasticity of the legs for efficient locomotion. Many design methods for robotic legs have been developed and investigated to mimic animals' efficient mechanism. In these methods, the robot's body is simplified as a muscle point and the leg as a spring. This simplified model is called the spring-loaded inverted pendulum, SLIP model. The spring constant is designed as stiff as that of an animal's leg of the same mass. RHEX is the pioneering and great example, featuring C-shaped flexible legs. Now I will talk about the purpose of the study. This study proposes to add a torsion spring in the SLIP model to increase the propulsive force by designing the torsional and linear stiffnesses appropriately. In this paper, the newly added torsion spring is called leg torsion spring, and the spring inherited from the SLIP model is called leg linear spring. The illustration describes how the torsional spring can produce propulsion force. In A, the leg touches the ground, the torsion spring is compressed, and elastic energy is stored in it. In BC, the elastic energy is released, which in turn pushes the body forward. Now I will explain how to achieve it. I mean, propose the methods. We assume that by designing legs so as to kick the ground strongly backward and horizontally due to elastic force, the robot can achieve a large propulsive force. For this, the legs must store a large amount of elastic energy just before they kick the ground. For the elastic force to push the body forward, the legs must deform in such a way that the toes project forward, as shown in figure A to C. To realize this deformation, here we propose a D-shaped leg. We designed the D-shaped leg by combining a C-shaped leg similar to the RHEX legs and I-shaped beam. Now, let me move on to the simulation model. The D-shaped leg is designed by a combination of the physical simulation and optimization with the following two policies. Policy 1 is to maximize the elastic energy stored in the leg just before kicking the ground. Policy 2 is to adjust the stiffness of the leg linear spring based on the mass of the robot as well as RHEX design. For physical simulation, entire D-shaped leg is modeled as torsion and linear springs. This is a 2D simulation model of the D-shaped leg. In this study, we perform a static simulation that reproduces the deformation just before the leg kicks the ground. To perform the simulation and optimization, we used Rhinoceros 6, a 3D CAD system, and its plugins, which includes a physical simulator and optimization functions. The optimization was performed using a multi-objective genetic algorithm named Octopus. Please look at this figure. This is the Rhinoceros screen. We defined the y-axis and x-axis as shown in the figure. The model was fixed at the top 2 points and 0.7 newtons was applied to this point as the normal force from the ground. The plots in red show the D-shaped leg before deformation. The plots in green show the D-shaped leg after deformation. The stiffnesses of the torsion springs in each C-shaped and I-shaped part are used as variables in their optimization. The objective function 1 was set to maximize the elastic energy stored in the leg after deformation. E is the sum of the elastic energy stored in the leg. K theta is the stiffness of the torsion spring. Delta theta is the angular displacement of each torsion spring. KL is the stiffness of the linear spring, and delta L is the length change of each linear spring. The objective function 2 is to achieve desired leg linear spring stiffness proposed in our hex. In particular, we set to minimize the difference between the y displacement of the leg's toe and the desired displacement. The desired displacement was determined from the stiffness of the appropriate leg linear spring. Here is a video of rhinoceros and its plugin while running an optimization algorithm. First, the stiffness is changed by an optimization plugin. Then a physical simulation is performed. Finally, the deformed shape is displayed and the optimization plugin evaluated. This process is repeated. This right plot shows the progression of the multi-objective genetic algorithm actually used for the prototype. The C-shaped and I-shaped parts are fabricated so that they achieve the stiffnesses by the optimization. Here, I will show you the prototype with 6 D-shaped legs, whose stiffnesses are defined by the optimization. We also fabricated the C-shaped legs to compare the locomotion performances with the proposed method. The comparison was made by changing the legs. Each of the 6 DC motors fixed to the body rotates one leg to perform locomotion. The 6 legs are divided into 2 sets, red and blue, and each set is synchronized. The phase difference between the red motors and blue motors is 180 degrees, so the red and blue motors rotate anti-phase manner. There are 4 essential parameters for a tripod gate. The working cycle is TC seconds. There are 2 phases in the cycle, the first phase and slow phase. In the first phase, the motor rotates the leg by 2 pi minus pi s radians during Tc minus Ts seconds. In the slow phase, the motor rotates the leg by pi s radians during Ts seconds. When phi O is 0, the range from this angle to this angle corresponds to the slow phase. Otherwise, the angle of the slow phase is shifted by phi O. In this study, Tc was set at 0.76 seconds. Also, Ts was set at 0.38 seconds. Experiments were conducted for different values of the control parameters, phi s and phi o. The control parameters of previous studies are used as a reference for control of the prototype. Now, I will show you the highlight data of the experiment. This video shows that the D-shaped legs increase the locomotion speed. In this experiment, the prototype moved for 5 periods, which then the distance traveled was measured. The speed was obtained by dividing the distance traveled by the elapsed time. We repeated the experiment 3 times with each parameter setting and obtained the average value. Now I will show you the experimental results. We tabulated the experimental results with various parameters. Table B shows the locomotion speed of the prototype with the D-shaped legs, the proposed method. Table C shows the locomotion speed when utilizing C-shaped legs, the related method. Horizontal axis is phi s, and the vertical axis is phi o. The units are meters per second. Table A shows the ratio of the proposed leg speed to the C-shaped leg speed. The units are in percentages. Greater than 100% indicates that the proposed leg speed is higher than the C-shaped leg speed. The results show that the D-shaped leg increased the locomotion speed by up to 89%. In the previous video, I showed you the experiment with this parameter setting. These are snapshots of the prototype during locomotion. The red colored legs are the state just before kicking the ground. These figures show that the D-shaped leg deformed as intended in the design phase. Due to the elastic force produced by this deformation, the D-shaped legs seem to have increased the prototype's velocity. Finally, I will conclude this presentation. We propose the method of designing legs to improve propulsive force through the use of D-shaped legs. In this design method, the stiffnesses of the legs are determined by simulation and optimization. In the future, we want to add distribution to the stiffness when running the simulations and optimizations. We will also change TC and TS and compare the moving speed. Thank you for your kind attention.
Thank you very much for this fantastic talk. We have time for questions, 2 questions maybe. I will maybe start with one question. So is it possible actually to estimate the energy that you can save by the approach you're using compared to the C-shaped leg? So how much energy you can save by this approach? You say that it was faster.
V-shaped leg increase the locomotion speed.
Maybe it would be interesting to also to look at the not only at the speed but also that the energy efficiency and there. Yeah, nice.
I think I can estimate it by using the deformations from the simulation.
Great.
Yeah, I've got a question. So I guess looks like there's some energy storage in return. Do you think that there might be also increases due to just maybe better ground contact and friction? It just seems to kind of mold with the ground better. That question makes sense.
Yes, I think so.
Great.
I would like to find a way to give it another function.
So if there are no other questions, I would say we move to the last speaker in this session. Thank you very much again for your talk. And now we will have Kevin Dye from the Biohybrid and Organic Robotics Group at the Carnegie Mellon University presenting Slugbot, an Aplysia-inspired robotic grasper for study and control. Please share your screen and start the presentation.
Hi, can everyone see and hear the presentation?
Yep.
All right, awesome. So yeah, I'm Kevin, and this is Michael, and we're going to be talking about Slugbot, which is an Aplysia-inspired robotic grasper for study and control. So the robot is inspired by Aplysia californica, which is a sea slug, and this model animal was selected for this project because it can exhibit multifunctional feeding behaviors, which we'll talk about in a second. But it can create these multifunctional feeding behaviors using a single musculature. And more importantly, it's been extensively studied. So the neuromechanics of this animal are relatively well known. And this enables us to have informed design principles when creating this bio-inspired robot that is based on Aplysia. More specifically, we're going to be taking a closer look at the Aplysia buccal mass, which is responsible for feeding. And the feeding behaviors that we're going to be looking at are biting, which is an attempt to grasp food when the animal detects a chemical stimuli. There's also swallowing, which is when the animal successfully grasps and ingests the food. And lastly, but not shown here, is rejection, which occurs when the animal expels inedible food from the buccal mass. All three of these behaviors have been demonstrated in a prior simulation. This is a one-dimensional neuromechanical model. You can see a representation of the kinematic elements here, as well as the Boolean neural model that incorporated some of the known neural connections within the animal. So in this simulation, it was able to create both all of the rejection, biting, and swallowing behaviors seen in the animal. And other previous work in bio-inspired Aplysia-based robots, this soft robot used pneumatic actuators to replicate the swallowing motion within Aplysia using peristalsis to push a spherical odontophore representation from one end of the robot to the other while grasping onto synthetic food. And so if we take a closer look at each of these prior works, the benefits of having a model and an in silico model is that it uses the neural-inspired control to generate multifunctional behaviors. And this enables the model to be used to test biological hypotheses in silico. But because this is a one-dimensional model, it lacks some of the real-world physics that a real physical system and animal might have. And so if we compare this to the in roboto model, the benefits of having a robot model is that it's inherently physical, inherently has three-dimensional real-world physics, and also incorporates some bio-inspired actuators that we might be interested in for bio-inspired robots. But the downside of having this in the previous work is that it lacked a multifunctional neural controller, and also lacked some of the key muscle groups that we want to investigate within the buccal mass. In addition, there was no kinematic data comparison between the animal and the robot. So the question that we ask ourselves is, how can we address the shortcomings between these two models? And to go about address this shortcoming, we wanted to try to take kind of the best of both worlds from these two previous approaches, and we combined those in our SlugBot, which is a slug-like uniaxial grasping robot. And so on one side, replicating the previous physical model, we have this bio-inspired mechanical system that is being controlled by a modified version of the previous Boolean neural controller, as well as a separate pressure controller. When we were looking at the design of SlugBot, we wanted to make sure we were basing it on the morphology of the apposite buccal mass, and particularly incorporating key muscles that have been identified to be important towards the different behaviors that we were looking at. So in particular, we wanted to incorporate I3 rings, which have been shown to be the major retractors of the system, as well as I1, which is a part of an I1, I3 muscle complex. So again, involved in retraction and changing the overall shape of the buccal mass. We also wanted to incorporate a distinct I2 muscle, which has been previously shown to be the major protractor of the system, as well as a deformable radular surface. And this radular surface is what's actually doing the grasping in the animal, and the soft mechanics of that system have been shown to be important to grabbing food. The rest of the system is more of a phenomenological model. So in the animal, the internal grasping structure of the odontophore is soft, in ours it is rigid, and we have analog muscles that replicate behavior but do not have direct morphological equivalents in the animal. So to construct the soft robot, we used soft pneumatic artificial muscles. Some of these actuators were based off of the McKibben design, which utilizes a braided mesh laid over a inflatable latex balloon and then secured to some barbed pneumatic fittings. These actuators, when they're inflated, they will contract along the primary direction and expand along secondary directions. But we're not just limited to making linear McKibbens. We can also create toroidally shaped McKibbens. So as you can see here, when inflated, this toroidal McKibben will significantly decrease its inner diameter, which makes it suitable for generating peristaltic motion. But not all actuators on a robot are McKibbens. In particular, the I2 muscle within the animal is a thin sheet-like muscle that is also somewhat Y-shaped. And so to maintain this flattish morphology, we look towards using flat pneumatic artificial muscles. We constructed a Y-shaped array of these muscles. And the way that they work is that when they're deflated, they are relatively thin. But then when inflated, they similar to the McKibbens, they will contract along the primary direction and expand along secondary directions. And so these utilize a reinforced elastomer design with an internal air cavity. To fabricate this muscle array, we used a liquid elastomer injection process where we poured liquid elastomer into a mold and also added in some additional elements such as a reinforcing polyester film. This was pre-cut to direct the contraction direction of the muscles. We also include a dissolvable film layer that will provide air channels for inflation later. Another important component to our robot is the odontophore representation, which we fabricated using 3D printing to create a rigid shell, as well as two grasping radular halves. These radular halves can open and close to grasp onto an object because they're actuated using some McKibben actuators. There's also a soft element on the outside of these grasping halves, which represent the radular surface within the animal. We have also included some sensors in this odontophore, such as an accelerometer and a force sensor. So these will provide feedback to our control system. And then moving on to the control system, it incorporates both a modified version of that previously mentioned Boolean neural controller, as well as a separate pressure controller that directly controls the muscles. So the Boolean neural controller takes in sensory feedback from the different onboard sensors, uses known connections and phenomenological connections in this neural circuit to generate motor patterns, which are then fed to the pressure controller to activate the different muscles. When we were going about transferring the previous in silico model to the robot, we needed to incorporate some additional connections to account for muscles that were not originally included in the biomechanics model. In particular, we needed motor neurons for I1, as I1 was not included in the previous model. And similarly, we needed motor neurons associated with opening of the odontophore. Previously, only closing was incorporated. And we also needed to incorporate spatially varying activation of I3. So in the previous model, I3 was activated en masse. but to achieve the necessary kinematics needed in the robot, we needed to incorporate a spatially varying activation of the I3 rings. And so we're able to do this by incorporating both known connections in the animal as well as phenomenological connections based on the robot morphology and behaviors in the animal. So in particular, we incorporated the B43 and B45 motor neurons, which have been shown to activate I1, as well as the context-dependent B44 and B48 opener neurons, which actually activate two different muscle groups in the animal, but in our robot activate a single motor group. And the interneurons of these motor neurons are phenomenological connections based on achieving particular behaviors. The B3, B6, B9 motor pool, which is shown to be particularly activated in retraction, was broken up into two subgroups, which we have labeled retractors unit 1 and then retractor unit 2, 3. And this allowed us to take the same interneuron logic for B3, B6, B9 and break it up into a spatially varying activation of I3. And then in addition, we also incorporated B10, which has been shown to activate the posterior region of I3 as it is hypothesized to be a context-dependent protractor in the animal, and allowed us to incorporate the posterior region of I3 as a protractor when our I2 reached the kind of end of its kinematic range. Moving into a video of the slugbot swallowing. So in this kind of mock feeding experiment, you'll see coming from the right tube that is simulating our food, as the adonophore, which is covered in a silver material, reaches a peak retraction, the gradular halves will close and it will grab onto the food. And then once it has successfully grabbed onto the food, it will begin to retract and pull that tube through the lumen. And as you can see, this is a real-time video, and so it is like the animal moving quite slowly, but it is successfully able to pull the tube through the lumen of the robot and deposit it between the halves of the upper halves of the Y of the I2. In the animal, this is where the esophagus would be, so it has something in the back to grab onto the food. This is something that we don't have in the current robot model, and so the role of the esophagus is being played by a human actor. Looking at the robot kinematics, we're able to do comparisons both with the previous in silico kinematics as well as the animal data. In biting, we can see that there's good phase alignment between the robot and the in silico model, as well as decent phase alignment with the animal data. In translation, when we look at the rotation of the adonophore, you see that again, there's good phase alignment, but there's a pretty dramatic difference between the magnitudes of the rotations that we're being able to see, where the robot has much lower rotational range in biting. And this kind of leads to one of the kinematic limitations of this model as a model for aplysia. Looking at the swallowing data, which was previously shown in that video, we can again see good phase alignment between the in silico and in robusta models, as well as good alignment with animal data showing this cyclic motion. And actually in rotation, we can see much better alignment between the animal and the robot data, both in magnitude and in phase alignment. Here you can see some of the neural activation commands that were used to control the kinematic model and the in robusta model. So on the left-hand side, these are the expected neural activations per cycle within the simulation. And on the right-hand side, you can see that the durations of these neural activations in the robot are significantly longer than in simulation. And this is specifically for biting. If we look at swallowing behavior, then we get a much better qualitative comparison in the neural activation durations between the in silico and in robusta models. So for our future work, we want to further improve slugbot by adding additional biomimetic elements. And our hope is that by including additional biomimicry, we can later use this robot for testing biological hypotheses. So as part of this, we want to integrate multifunctional switching between the 3 primary feeding behaviors. To accomplish this, we also want to implement object identification using some of our sensors so that we can successfully implement rejection as long as the robot can identify what object is grasping onto. As Michael mentioned, right now we are using a human actor to substitute the effects of the esophagus. So we want to improve our object grasping and handling. And lastly, we want to eliminate any use of external sensors. So we want to transition towards solely internal proprioceptive sensing. So yeah, we'd like to thank everyone who made this possible. And are there questions?
Thank you very much. for this very nice presentation. I introduced only one speaker. Maybe if the second speaker just can say his name.
Hi, my name is Michael Bennington. I'm also a student here at CMU.
Perfect, thanks. So please ask questions. We'll start maybe with the questions regarding the size of the objects that you can grasp. Is there, or size and shape of the objects?
Yeah, so for these preliminary tests, Kevin's going to go back to the video. For the original tests, we were looking at replicating similar tests that were shown in the previous Mengen robot, which was grasping small tubes, so akin to smaller sizes of seaweed. As we move into a more deformable and more biomimetic radular surface, we hope to test this with different sizes and different shapes of materials to show that this soft deformable surface is good at both multifunctional in terms of what behaviors it can perform, as well as being able to grab a different variety of foods using the same musculature and neural circuits. So here, the tube is fairly small compared to the robot, but next steps will be to test on a variety of different sizes.
Okay, thank you. Is there a question from Roger Quinn?
Great talk. I love the McKibben actuators. You've done some novel things with McKibben actuators. Have you done life cycle tests with them to see how long they may last before there may be a fatigue failure or something?
Yeah, so we did experience that during the testing and experimental phase of building this robot. The initial McKibbens that we constructed lasted about, let's say, maybe 10 to 20 hours of testing. And then after that, some of the experimental McKibbens that we added later tend to have shorter lifespans, potentially because we were stressing them more and fatiguing them earlier. And we found that this is mostly due to rips or tears in the latex balloon itself. So we're going to be looking into ways of either reducing that chance or finding ways to repair them relatively quickly.
Okay, there's another question from Falk.
Yeah, thank you for this beautiful talk. It's really awesome to see this robot in action. I wanted to ask, and all the other questions I had were already asked, how much force and how much pressure are you actually using? And are you using the same pressure for the Y-shaped actuator as for the McKibbens, or are different pressure sources?
Gotcha. Yeah, we tried to match up the range of pressures to be relatively comparable between the Y-shaped flat artificial muscle and the McKibbens. What we found was that somewhere in the range of 10 to 15 psi was the maximum for the McKibbens that we applied, and then for the Y-shaped muscle we stuck around the 8 psi maximum.
Mm-hmm. Cool. Thanks.
And there's actually an internal question, but
so I might have missed this, but for the kind of impact of this, is this more like a representation studying how you can kind of take this kind of biological system and represent it in a physical way, or is it more like a goal to make a better grasping system or another grasping system that might be implemented in X field or X?
Yeah. So this specific robot leans more towards the biology, but the goal is for this to be slightly branching. We can look one way, let's make this more biomimetic, make this an appropriate test platform for neuromechanics, as well as this slightly able to grasp a whole variety of different things, react to the environment. How can we take principles from it and apply it to designing better graspers? So a little bit of both.
Okay, so if there are no further questions, there's another question. Please go ahead.
Yeah, my question is that what is the role of the rate part, this C curve? at the back. Is there any role of this kind of structure?
The fin structure was represented by the red in the cartoon.
At the backmost part, the C-type curve. Okay, so in your animation.
Yeah, so I can go back to the design of the robot. I think you're talking about this particular I2 representation of the muscle.
Okay. This is kind of I2 things, right?
Yeah, it acts as a protractor during the feeding behaviors. So it'll push the odontophore forward to start protraction.
Okay. And do you have any comment on the size of I2 or I3 or radius of this odontophore? Do you want to comment on that? Is there any kind of order in the size to have this kind of mechanism or to have this kind of movement? Do you have any observation? Yeah.
Some of the sizing of these components, well, one for the odontophore, this entire robot is significantly larger than in the animal. Mostly because of the actuators that we're working with and the limitations of constructing the odontophore. But as far as sizing the muscles relative to each other, in particular with the I2 muscle, we're kind of limited by the amount of contraction that these artificial muscles can provide because they don't contract as much as biological muscle. So we tried to get a ratio that's backable by what we find in literature, but we need to further make improvements to this I2 by maybe making it more elastic so that we can make it smaller in the future.
Okay, thank you.
So I would say we can thank all speakers again for their nice presentations. Now we have a coffee break of 18 minutes and then are starting the short talks. See you later.
Hello, welcome back. Okay, so I'm gonna give this a go. So I'll wait like maybe 30 seconds, 1 more minute. But this is the short talk portion of the day 1 of the conference. We have 10 pre-recorded short talks. And then we'll open up the floor for questions. Hopefully, you know, all the authors are here and all the presenters are here. And let's let me go ahead and play this.
So we don't have any sound.
That's what I was worried about. Okay, hold on. Let me try one more thing.
Hi everyone, and welcome to my short talk. My name is Stefan Konrad, and I'm a PhD student in the Cluster of Excellence Litwatz at the University of Freiburg in Germany. As we all know, soft growers have a great potential, but what is still limiting the speed of their development is the easy access to a suitable and low-cost rapid prototyping technology. FDM 3D printers have already proven their potential for the creation of soft robotic elements, but a still ongoing challenge is the combination of materials with different mechanical properties. In order to address this, we have developed an innovative multi-material 3D printing platform and presented it on the conference in 2020. It is able to perform on-demand tool change on runtime, what means it switches between different tools which are completely independent and loaded with different filaments. Using this device, we are now able to create this soft robotic arm, which is shown here schematically in this illustration. Here in red, we see an arm-like structure made of PLA with a compliant hinge here, and in green, a pneumatic actuator made of TPU A70. Both pieces are connected by these joints here, which can rotate freely. And since the actuator is a bit smaller than the arm structure, it is located here on the supporting PLA platform. Since the TPU printhead needs some print time to reestablish a consistent flow rate after it has been in the standby position, we placed this prime tower here right next to it, which is always printed first, and then the printhead goes to the actuator. Here we see it in real life. And in this diagram here, we can observe the print times. Color marked, we see in dark green here the actuator and the arm in red. They took about the same print time. And here in light green and blue are the tool changes. We see that prime tower and tool changes consumed about 30% of print time. Since there's no support material which has to be removed from the actuator, we could directly take it off a print bed and connect it to our test stand. In the first step, we did not apply any pressure to the actuator, but we loaded the arm with a weight of 270 grams. And as we see in the picture here, the actuator is extended and the arm bends downwards. In the second step, we additionally applied 1 bar overpressure to the actuator. It extended even further, and we can see the joint rotating. Finally, we evacuated the actuator, and the arm snapped upwards, lifting the weight of 270 grams. That's it for a moment. Our next steps will be a further characterization of the soft robotic arm, an optimization of the print parameters, and of course, design and creation of even more complex soft robotic parts. Thanks for your attention, and thank you for considering our paper for the conference. And if there are any questions, please feel free to ask in the Q&A session later.
Hello, my name is Emmanuel Andrada. Together with my colleagues, Andreas Carbot and Martin Fischer, I'm very happy to be here at Living Machines 2022. The new next project, C3MS, we try to understand how extremities communicate with low-level control centers in the spinal cord and their interactions with high-level control centers in the brainstem in small mammals, in our case, hyena. As you may know, to infer dynamical in our case, neuromechanical control strategies implemented by any system or animal, it is necessary to characterize how they respond to external perturbations. Biomecanicists use passive perturbations, like you see here in this case, sudden drops or steps, in order to mimic as far as possible animal locomotion on rough terrains. We think, however, that in order to infer interactions between spinal reflex control and higher locomotive centers, reproducible active perturbations are necessary. Therefore, together with the industry, we developed a 3 degrees of freedom actuated platform we call the Shaker. The Shaker was designed to generate single or combined horizontal, vertical, and tilting perturbations with oscillation frequency up to 10 Hz. We also integrated force plates into the platform in order to measure ground reaction forces. With this device, we can measure active perturbation during standing and walking using biplanar X-ray fluoroscopy, implanted tantalum beams, implanted EMG in order to measure muscle activation, and we measure also single leg ground reaction forces. Let me show you one of our preliminary experiments in which we have an active sudden drop occurs during a rat string locomotion. Note that we have recorded ground reaction force before, during, and after the perturbation. We expect that by measuring motor response to a range of perturbations, we can define the functional mapping between deviations in limb states, center of mass dynamics, and muscle activations. Just want to thank you to our sponsors and all the people who worked in the design and construction of the Shaker, development of the experimental setup and the experiments, and to you, I want to thank you for your attention.
Hello, my name is Heiko Stark. I come from the Friedrich-Schiller-Universität Jena. I would like to briefly introduce our extension of an existing dog model. Why dogs? Dogs are an interesting object of research because evolutionarily very different breeds have been bred within a very short time. This concerns their size, shape, and behavior. For this reason, we have assembled a bigger model which includes the bony structures and the muscles. This can be read in the publication listed here. Of course, a single model will not reflect the full range of variation in dog breeds. So our goal was to create more breed-specific models. To create the new musculoskeletal models, we choose the Dachshund and the Great Dane. Both represents extreme breadths in terms of body size and the relationship between body length and width height. However, other breadths are also to follow. These models can then be used to study breed-specific problems, such as rupture of the cruciate ligament or dysplasias of the skeleton and connective tissue. To create the new models, we use the scaling function of OpenSim. This allows the individual segment masses, inertia, length, and muscle parameters to be scaled uniformly or individually for all 3 axes. The maximum muscle force, on the other hand, must be scaled manually. The scaling presented here are only a first step towards a tool for locomotion and health analysis of dogs. Further steps are necessary. For example, the joint actuators for each model need to be optimized as well as the models themselves validated. We are confident that these additional models will contribute to the analysis the influence of body size, physical mobility, and disease on neural control and joint loading during locomotion in dogs. Thank you for your attention.
Hello, I'm Hemma Phillimore, and I'll be presenting our paper Gut Feelings, which was produced with my colleagues Martin Garrett, Martin Grauel, and Max Jones. Our paper is about artificial personality generation for robots. We introduce a new concept, the use of a bioelectrical signal source as a core component of the personality generator. Our hypothesis is that by including this bioelectrical signal, humans are more likely to accept and trust the robot companions they interact with. This diagram shows an overview of our personality generator. The generated personality, outlined in red, comprises personality traits and mood. The contribution of this work is outlined in blue. We investigate a system where human interaction through verbal exchange, combined with a bioelectrical signal, determines the generated personality. The final part, the embodied actions that the robot exhibits as a result of its personality, will be explored in future work. The inspiration for this work was the gut-brain axis, a bidirectional neural communication channel which links emotional and cognitive centers of the brain with the gut. It has been associated with relating the gut microbiome composition to human personality expression. The bioelectrical signal comes from a microbial fuel cell, or MFC, a two-electrode system that generates electrical power from oxidation of an organic substrate via microbial respiration. The electrical power produced increases with the available energy of the substrate within the MFC. We can think of an MFC as a biological battery or artificial gut. By using this artificial gut within the personality generator, the robot personality is sensitive to not only the robot's external environment, but also the robot's own perceived physiological requirements or internal state. We adopt a widely used approach for categorizing personality, the Big Five personality traits. We set the goal of designing a personality generator that achieved three aims: believable and varied personalities, predictable and unexpected behavior, and history dependence. We then devised a simple model to take the voltage from an MFC as an input and generate a personality that met these criteria. The voltage from an MFC is shown as a black line in the plot on the left. The boundaries between four bands, A to D, were defined using the mean and standard deviation of all data points in the voltage time series. The rate of change of the MFC signal was also plotted, and four bands E to H were defined in a similar way, though this plot is not shown. At regular intervals, the boundaries between the bands were updated, and the ratio of data points that fell into each band was used to define a happiness score and a stability score. The happiness and stability scores were used in combination with so-called order and answer scores, which reflected the quality of the robot's verbal interaction with a human, to calculate a score in the range 0 to 1 for each of the Big Five personality traits. The temporal evolution of the Big Five personality traits generated from the MFC voltage is shown in the plot on the right. The natural variation in the microbial population causes electrical output to vary between otherwise identical MFC systems, producing different distributions of the Big Five traits for each MFC. This satisfied our objective to generate believable and varied personalities. The MFC signal satisfies our objective of combining interpretable and unpredictable behavior, as the response to each fuel replenishment was a peak in voltage. However, the amplitude of each peak varied significantly. Finally, the personality evolution is history dependent in that the current personality trait distribution depends on all preceding data points in the time series, but also in that the MFC voltage output is affected by factors such as population growth of the microbes within the MFC. Future work will further this early stage investigation that introduces the concept of a biohybrid system for robot personality generation.
Hello everybody, welcome to my presentation about the development and characterization of a soft bending actuator. My name is Armin Jamali from the University of Freiburg. Artificial muscles made of soft and compliant materials are implemented in a variety of fields such as soft robotics, medical tools, and haptics. In any case, and independent from the type and design of such actuators, the choice of material plays a critical role for their performance. Dielectric elastomer actuators, DEAs, also known as a subset of artificial muscles, are made of thin elastomer layers embedded between two layers of soft electrodes. In the journey of finding a suitable elastomer material with relatively low elastic modulus, high dielectric constant, and high dielectric breakdown strength, we found the EcoFlex 0010 mixed with silicone thinner as a promising candidate. For ease of referencing, we call this compound EcoFlex 10P. In this study, first we investigate into the mechanical and electrical properties of this silicone-based elastomer. Then we explain the fabrication method of a soft bending actuator. So here are the characterization results. For the electrical breakdown strength, we tested 271 micron layers of EcoFlex 10P for multiple specimens, and it resulted in 21 0.9 volts per micron. This value is important to know how far we can increase the voltage to reach to the maximum strength. The results of the dielectric constant are also shown here. It's 4.4 at 20 Hz. For the elastic behavior for multiple specimens, we measured 0.052 megapascal for strains more than 500%, and we got 0.01 megapascal for strains lower than 300%. The elastic modulus and dielectric constant of EcoFlex is very favorable compared to Stilgard silicones, as you can see here. Finally, to try this elastomer in an application, we fabricated a bending actuator by increasing the mechanical resistance on one side of the and making it bend in that direction. This was done by placing pieces of PMMA as backbone structure on one side of this actuator. Then it was hanging vertically in front of the laser camera to detect the bending profile. Here we can see the results under 0 to 8 kilovolts, bending and lifting its own weight. Thank you very much for your attention. I'm looking forward to your questions and suggestions in the Q&A section.
Hi, I'm Shmuel Patel, and I'm here representing three of my Jenny, Vivek, and Nick to talk about how we developed a robotic platform for the testing of a simple stereopsis network. The reason we undertook this task is simply that visual systems use a lot of computing power. Through their real-time nature, as well as implementation of deep neural networks or artificial intelligence. This often results in design limitations due to the large GPU and CPUs, as well as batteries required to operate these systems. We believe that insects can be used as inspiration, however, to fine-tune visual systems due to their low resolution, yet ability to detect colors, lights, and patterns at an adequate level. First, when handling this task, a neural network was developed. And the network works by first breaking the field of view into simple bins, reducing computational complexity. Then, the two field of views of the cameras are analyzed together to see which corresponding bins were activated when an object passed through the field of view. Then, once the corresponding bins are identified and synaptic weights are applied, a distance can be found by identifying which neuron was activated to the highest extent. Then a robot was developed to implement this network on. The robot featured two Pixie 2 cameras, which provide front-end processing to identify the location of specifically colored targets, a Raspberry Pi 3 to do the onboard processing, and a Zumo chassis and Arduino to do the mobility aspect of the robot to maneuver. Then, once these were all constructed, some preliminary work was done to demonstrate that the two Pixie 2 cameras will have interocular disparity, simply that the left eye and the right eye see objects differently. And you can see from the two pictures on the right that the left eye and the right eye do see the objects from a different viewpoint. Then in the future, we hope to integrate the network and the robot more effectively and show that it works in a more robust environment rather than just simply a controlled visual environment as employed in the lab. Finally, we want to utilize the network to provide outputs to the robot, to tell it to turn left or turn right based off the location of an object in the field of view. We enjoyed working on this project, and we are very excited to work on this project further. And I hope you enjoyed my talk today. Thanks for your time.
Hello, everyone. My name is William Norse, and I'm a PhD candidate in the Biologically Inspired Robotics Institute under Dr. Roger Quinn at Case Western Reserve University. Today, I'm going to be talking about the SNS Toolbox, a tool for efficient simulation of synthetic nervous systems. Over the years, our lab has worked on developing synthetic nervous systems, which are control networks comprised of simulated dynamic neural networks, and we've used these to control simulated and real robotic hardware. Currently, all of our existing neural controllers do not run on board our robots. They're tied to external PCs for control. We want to move to a software ecosystem for running our controllers on board the robots. So this system must be capable of conductance-based modeling, real or faster than real-time interaction, and no forced tie to a GUI interface. We'd also like to be able to just run larger networks in general. So we looked at existing software solutions. And although Nengo comes close, none of the solutions quite come to what we need for running our synthetic nervous systems on board real hardware. So we chose to implement our own software suite, which we call SNS Toolbox. SNS Toolbox focuses on simulating the specific neural dynamics we're interested in. For our full methodology, please consult the paper. It consists of an easy-to-use Python API with both CPU and GPU hardware support. It's available now, open source on GitHub. There's also a full suite of documentation, including instructions on how to install the package, various tutorials on how to simulate and design networks, and a full API reference. We also have a quick software demo. So we take a small input image, small so that we run this quickly in the video. And we're going to do a two-stage visual processing pipeline on this image. So we're going to convert the image into a neural retina implementation, and then we're going to high-pass filter this image onto a lamina population. This is based on high-level functionality within the insect optic load. So we're going to create a general network. This network consists of over 2,000 neurons and over 8,000 synapses. And this is going to be implemented for network construction in only 18 lines of code by defining two populations and the pattern of connections between them. We then set up the simulation and compile the network to a simulation backend. And then we'll run the network in a loop for 3 time steps. And at the end, we can see that the lamina output converges to a high-pass filtered representation of the original input image. For future work, we'd like to increase the level of optional complexity available in the neural models for our system, as well as adding wrappers for popular simulation software such as PyBullet and OpenSim. I'd like to acknowledge our sources of funding, as well as all of my references. Thank you very much for listening.
On behalf of Case Western Reserve and Virginia Tech, I'd like to present to you animal acceptance of an autonomous pasture sanitation robot. Runoff from animal waste contaminates water supplies and decreases pasture health. Mitigating the negative effects of animal waste requires either significant manual work or reducing pasture capacity. Autonomous management of manure via in-pasture systems could greatly reduce these negative effects. However, no such systems currently exist for in-pasture automation. This work constitutes the first steps towards this kind of autonomous management. To do so, we introduce a robot to horses and cattle under controlled conditions to observe animal response. Specifically of interest were how animals showed distress or acceptance of the robot, and how the approaching robot was treated, and then we began to outline regions where a robot can coexist with livestock while minimizing stress. The robot platform used for this study, shown here, is termed PSR1. PSR1 is a 4-wheel drive skid steer robot with an overall volume of about 1 meter cubed. PSR1 is equipped with an onboard Intel Skull Canyon operating ROS Melodic. For a sensor payload, PSR1 is localized via a U-Blox RTK GPS and uses an Ouster OS132 3D LiDAR to record the world around it. Animal trials were conducted on cattle and horses. Cows were split into 2 herds of approximately 15 cattle each, while horses were split into 3 groups between 5 and 8 horses each. Each trial had 4 phases. First, a 5-minute motion phase where the robot moved in a random path in the pasture. Next, there was a first stationary phase where the robot halted all motion for 10 minutes. Following the stationary phase, the robot approached the nearest animal until that animal fled. Finally, the robot again ceased all motion. During each stage, we measured the minimum distance between animal and robot, as well as the number of interactors, animals which made contact with the robot, and the number of interactions, the number of times an animal made contact with the robot. Contact refers to animals approaching within 25 centimeters of the robot. There was a high variance in animal behavior for both species. However, there were a few trends. Both cows and horses interacted more frequently during the stationary phases. Cows never made contact during the motion or flight phases. Meanwhile, horses interacted more frequently and did so even during the motion or flight phases. Both species interacted with the robot after the flight phase, showing that they weren't overly stressed by the robot. Animals of both species made contact with the robot before and after the flight phase. Animal interactions for both species tended to increase during the secondary stationary phase, showing the robot flight phase did not introduce large amounts of stress. We obtained rough values for animal flight distances and interaction for each species. Cattle more consistently tended to flee, while horses did not. Robot behavior profiles may therefore need to be different depending upon species. Statistical analysis of animal behavior has low power due to highly varying behavior and a low number of trials. However, using values from these tests, we can synthesize new experiments to outline flight ranges and interaction metrics with greater statistical significance. Thanks for your time.
Hi everybody, I am going to present our work on quasi-static modeling of feeding behavior in Aplysia californica. I am Vidisha Kundu. I have done this work with Stephen M. Rogers and Gregory P. Saturn at the School of Life Science, University of Lincoln. Here is the video of the Aplysia feeding behavior. It is mainly biting, showing its mouth opening, and the part which is coming forward and going backward is called radula. Aplysia has been used for long decades for studying neural control. Here is the picture of anatomy. It is mainly the side view of the feeding apparatus. We can see the location of radula. Radula will go forward towards mouth and grab the food. It will come back and deposit the food near esophagus. Muscles will help radula in this movement. Here is the diagram of the original complete Newtonian second order model. We can simulate this second order model and we can generate the animation from the output of this system. But there is an issue in the original second order model. It takes too much time to simulate. So our question was, is there any way to reduce the computational time? In their model, they found the equation of motion from Newton's law, balancing all the muscle forces with the inertia. But we have observed that acceleration are very negligible for this system and it is computationally very expensive. Also, we have observed the energy storage for the muscle stretching are much higher than the kinetic energy. So we have done quasi-static reformulation by getting rid of the acceleration term and only concentrating on the muscle forces. Here we are showing how we can find our solution. The blue color forces is the positive protracting force which is taking radula towards jaw, and the red color forces is the retracting force which will take radula to take it back its original position. The profile of the forces is changing because of the muscle activation. After balancing these forces, we will get the static equilibrium point. We simulate both the model with this firing frequency function. Here is the output. The turquoise color curve is for second order model. Dash green color curve is for quasi-static model. They are almost same with few exceptions for small error. There is great reduction of computational time. It is almost 100 of the second order model. Thank you. If you have any question, let me know.
Hi everyone, my name is Dia. I'm a Tech and Math Treasures Lab here at Northwestern University. Today I'm presenting the work using Deep LabCut to predict locations of subdermal landmarks from video. This work is a collaboration between our group and Martin Fischer's group at University of Jena in Germany. So Deep LabCut has been widely adopted by many labs for position estimation of skin landmarks due to being highly automated and working well with video obtained from standard off-the-shelf cameras. However, studies that require highly accurate kinematics need to estimate the position of the skeletal structure itself, not the skin above it, since slippage between the skin and skeleton might lead to some inaccuracies. ETSROM is a method that uses high-speed X-ray cameras to obtain 3D dead accurate estimates of subdermal landmarks, but ETSROM is expensive, not many labs have it, and labs that do have to do manual annotation, which can be time-consuming. Here we present an approach that trains deep LabCut models to estimate dead accurate positions of subdermal landmarks just from video from 2 standard cameras. We test this approach by estimating skeletal and joint features in a locomoting rat's forelimb. So how do we train these deep LabCut models? We first simultaneously record a rat in an overground running task with 2 standard cameras and 2 high-speed X-ray cameras used for ETSROM. We then use ETSROM to obtain objective 3D points, and then project these points onto frames obtained from the live video cameras. We use these frames to obtain the 2D projected points as training data for camera-specific deep LabCut models. Then once our deep LabCut models are trained, we can record the rat just with 2 standard video cameras and use our trained deep LabCut models to predict the projected position of subdermal landmarks. These labeled frames from both cameras can then be triangulated to get predicted 3D points that are conceivably depth accurate. So how does this approach work? So we simultaneously record 4 trials of a rapid and overdone running task. We took the first 3 to train our deep LabCAD models, and in the 4th trial, we predicted 3D points just from our trained deep LabCAD models, and compare these to the objective 3D points obtained from the X-ray cameras. So here in A, we can see that the error in 3D position estimated by deep LabCAD remains reasonably low, just a couple of millimeters error. Perhaps more important, in beam C, we can see that the actual kinematics obtained from deep LabCAD matches the kinematics obtained from EdgeStorm quite well, with just a couple of degrees of error in joint angle. While this is preliminary work, we can start to see the power of this approach. Here in B, we can see in blue that the X-ray cameras capture a much narrower range of frames than the standard video cameras, represented in orange. While we aren't able to validate the accuracy of these DLC predicted frames outside the X-ray camera range, we can start to get a sense that this approach can be potentially used to get accurate kinematics, but across a wider, much more versatile range of experimental conditions. The real goal of this method, and the reason why we are pursuing this approach, instead of just applying deep LabCAD directly to help label X-ray frames, is that potentially if a model is generalizable enough, it can be used in labs that don't have access to X-ray cameras at all. Thanks.
All right. Excellent work, everyone. Thank you for your short talks. Now is the portion where we will open it up to any questions to the authors. So please go ahead. Yes, Vicky.
Hi, hello. Thank you very much for your talks. Very, very interesting. I had a question for the short talk number 5, gut feelings towards robotic personality generation with microbial fuel cells. And the question that I have is the following. It's not clear if this has been like actually implemented, or maybe I missed that part, if it has been implemented on an actual robot. And if it is not, I would like to ask the authors, how would they envision such an implementation for the generation of personality using the microbial fuel cells?
Thank you, Vicky. Maybe I can take that question. I was the person who gave the talk. So the work that's contained in the talk and detailed in the extended abstract was done during COVID. So it was done using existing experimental results from previous MFC experiments and then done as a sort of simulation. But we're currently in practice doing it using real MFCs and collecting the data from them in real time and having a sort of personality that evolves over time that's just sort of represented on a computer. I think going forward from that, what we'd like to see is something where a human can actually interact with that computational system and bioelectrical system by kind of feeding food or fuel, whichever you want to call it, into the MFCs and through that kind of establishing a relationship between the person who is like feeding and kind of caring for the robot and how the personality sort of develops. So it was done in simulation, it's being done in practice now, and then I guess that's how I see it being manifested in an actual robot.
So then you would envision some sort of bonding between the feeder in that sense and the robot, not just personality. So there's some sort of like need or some interaction that has to do also with survival, if you wish, in a sense.
Yeah, absolutely. So there's a sort of, yeah, I mean, the robot in that sense is kind of a dependent on the human anyway, however they refuel it, right? If you charge up their batteries or feed it some food in the way that we understand food, it's a similar relationship. So yeah, I guess it's putting it in a context of care or familiarity or a kind of dependence, as you put it.
Okay, it would be interesting to see that and also see how you might compare different kinds of approaches to see whether actually this is preferred over some other personality generation. So you could have a lot more, let's say, well-studied experiment. I
agree.
It's interesting. So it's very nice. Thank you.
Thanks.
Okay, Roger, I see your hand is raised. You're muted, Roger.
You usually like it when I mute myself. Anyway, I have a question about the modeling of different dog breeds on the basis of a validated model. It seems that the point is that, am I correct in thinking that it's like all dogs are basically, the biomechanics of dogs are basically the same, very similar, independent of breed, independent of size. Is that a conclusion that we can make here?
For the dogs, it's only a size change, but also a change in the muscles. So, for example, there was the bulldog that have very big muscles on the legs, and in contrast to that, the white one has more strange muscles, and so it has other body size and shape.
Okay.
So it's different. It's
So
not
they don't have the same dynamics or the same kinematics. They differ in form, and depending on the form, change the kinematic pattern and also some kinetic pattern as well. So that's why the idea is to show or to see how this mass and dimensions change influence yes, the dynamics of the joints and the work on the joints, and the possibility to infer some idea how yes, disease can be yes, developed because of a different position of the extremities or mass yes, different mass yes, conformations. So that was your question.
I think I understand.
Yeah.
Okay. Any other questions?
Well, here's Paul with a question. So on the SNS toolbox, so there are a number of these simulation platforms available for simulation of embodied systems and to link them with models of nervous systems and control. So can you say a little bit more about what you think is the unique contribution that this toolbox is making? What makes this different?
That's a very good question. So the main thing that we were looking for is there are a lot of tools out there. You're right. There's a lot of tools out there that focus on kind of enabling embodied neural systems. The thing with these is that they tend to be like, sort of example with the neurorobotics platform, you have a kind of a cloud client and you're using an integrated gazebo simulation and you're using the integrated scripting and all that sort of thing. And what that ends up happening is that if your particular, let's say if you wanted to run it on your own individual robot, it's very hard to remove all the extra components that you don't need from the neurorobotics platform to be able to use it on your own system. So what we wanted to start focusing on and kind of head towards is just make an engine that focuses on simulating the neural dynamics so that you can just pass in the inputs at whatever current state you're at and get what the state of the network is, and then use it as a component in whatever system you're trying to implement. So instead of trying to focus on building an all-in-one system that encompasses neural dynamics, muscle simulation, all that sort of stuff, just focus on doing the neural dynamics, and then for different applications that need different sub things, build tools that work with it to do that sort of thing. So we're saying that we're starting to work on wrappers for interfacing with simulators like OpenSim or PyBolt. That's not requiring you to use OpenSim or PyBolt. It's just saying, taking vector states of neurons, here's how we can use it to feed into these other systems. So it's a small use case tool.
Okay, but then to really make a contribution, either you give some sort of hyper powerful specification language that people can move forward very quickly, or right, you provide very efficient APIs to these other environments, so you can very quickly, tool up to run your experiments. So what's the strategy for you?
So we didn't focus on this in the video just because the video is pretty short. But essentially, this is right now, this SNS Toolbox is focused on these very specific neural dynamics. So it focuses on how do we simulate these specific neural dynamics in as efficient of a vector manner as we can. And also, a lot of neural simulation packages are focused, ones that can perform at scale, tend to be focused on networks that can be described in terms of layers. You have, one layer connected to another layer with a dense connection going down and down and down. Just trying to pick what could fit into the video, I didn't include this part, but the fundamental structure of the SNS Toolbox is it's designed for being able to simulate very complicated network structures with different levels of cycles within the network. So you can have essentially any neuron in the network can be connected to any other neuron in the entire network and simulated in that way. So it's less focused on these very large, deep networks. It's more focused on kind of smaller networks. Yeah.
Right. Well, but I can only recommend that indeed be always very clear about the unique selling point of the tool you build, right? Because it's a huge space, there's lots of things happening, and you can drown very quickly so that you have no traction, and people would think, well, yet another simulation tool, who cares, right? So I can see the purpose of this, but make sure you stay on mission, because it's a relevant thing you're doing, and it would be great when, of course, to get some traction also in the field. So, well, good luck. Great work.
Thanks.
Thank you, Paul. Thank you, William. Vicky, I see your hand is up.
Yeah. So I have another question regarding presentation number 5, animal acceptance of an autonomous pasture sanitation robot.
Yep.
Okay, great. So if I understand correctly, so then you have this robot that would do some sort of autonomous cleaning of manure or something like that, correct?
That's the ultimate goal, yeah.
Right. So the thing that you presented so far is a pilot testing of how the animals behave in the presence of the robot, whether when the robot is stationary or when the robot is moving. Right?
Yeah, and how that changes across those different regions of activity.
Great. So then my assumption is that you want these animals not to be stressed, and you would also, so there should be some sort of, if you would launch this system, you would want the robot to adjust its behavior given the behavior of the individual robots, in the individual animals. So because also from what I understood, there is some individual differences even between the differences of cows versus horses. So how would you have you envisioned to do that or implement that?
So I would envision that there would be on deployment of a robot, there would be selected for behavior profile. For instance, a robot might not directly approach something. It might try and kind of circumnavigate a little bit more broadly. It might be perfectly fine driving towards a collection of animals. And the relationship between those kind of behaviors depends on animal species, or it looks like it might, since this is sort of a preliminary work. So I envision that would be selected at deployment as opposed to updated during, but it's possible that it could be. It's a system that can or could conceivably adjust to, if it has a particularly interactive member in that herd, it can potentially update some of its, like, okay, well, I know I need to stay this far away, or I know I don't need to worry about that. So I consider this kind of plastic method change in the future.
Yeah, because I mean, okay, it makes sense to have either in advance some parameters that says this now is a cow, that the kettle is like the group is a cow or a horse. But I would assume also that you might want to have some distance regulation also for the safety of the robot, because maybe these animals might actually, I don't know, attack the robot. You might.
They certainly did mob the robot in trials.
There you go. There you
Yes.
go.
So
Yeah.
there are potential other applications or other things that we can go down to look at reducing unwanted behavior. Right now, the discussions of those are in sort of structural metrics, sort of like, how can I construct a robot so that it's less likely that they can come in and push the power button or the e-stop, or they can knock it around. We haven't had any instances where we've been anything more than inquisitively licked, but the potential exists in the future for us to kind of play around with, maybe sound or flashing lights of some sort to kind of warn away or play around with getting them away from the robot in that sort of circumstance. But we're kind of proceeding under the initial thought that they're going to navigate around, or it's going to be a kind of ignored feature of the pasture, and we're hoping to kind of play in that space. And how close can we get? How can we approach? How can we avoid animals in pasture?
Yeah, exactly, because you might want to modulate the speed. Also, I would assume that, yeah, I mean, because maybe right now the interaction was not long, right? It was short.
Yeah, the totals are like under about 20-25 minutes of total exposure. And that's primarily due to sorts of limitations in animal testing. These are pretty time intensive and resource intensive trials to set up. So we are definitely interested in doing longer term exposure testing with these animals.
Because this could easily be something like user adaptation, only instead of having it as a human, you have an animal as a potential user or interactive. So it would be interesting to see how this evolves. So yeah, thank you very much.
Thank you.
Thanks, Vicky. Thanks, Ian. I actually had a similar, along those lines, I was just thinking about it as you were speaking about it. I feel like there's two approaches to potentially take. One is the camouflage approach, where you try to go unnoticed and, hidden and, make the animals unaware, or you take the approach of kind of like, attempting to become one of them, or something that they are already familiar with. And I was wondering, if you went through that decision process, and if you considered, trying to behave or mimic something that's easily mimicked in some ways, but something that they're already familiar with.
I think the high level, like, ultimate form of this would be you just have an autonomous artificial cow mopping up a pasture. So I think that biomimicry would be the ultimate way to do it. However, because of the limitations in construction, the limitations in our robot designs, it's difficult to have a wheeled robot that the cows will interpret as another cow. So I think for the moment we're going to go more for the avoid, try and blend into the background, as opposed to like mimic and become one of the herd.
That makes sense. I was just curious. Thanks. Other questions for any of the short talks that we just watched?
I think Roger might rub off on me too much here. That autonomous robot looks a lot like the snowplow robot. Do you plan on using the same
know,
algorithms?
it does have a striking resemblance from that for some reason. As it turns out, I believe there are going to be a completely different set of things because the challenges are very different. From a low level, yeah, there's turn and drive and issuing goals and acknowledging navigation. But from the high level of how am I going to get there, it's going to be different. But yes, well spotted. That is in fact a reclaimed. It's an off-season snowplow robot that's pulling double duty.
Thanks, Alex. So I actually had a question on short talk number 9, the quasi-static modeling of feeding behavior. If Padisha is, oh, hi, Padisha. So maybe you went over this in your talk and I missed it, but I was, you made this huge improvement from a 14-second simulation to a 1/10 or a 100th of a second.
Yeah.
I was just kind of wondering if that's the extent, or if there's any reason to go beyond that and try to further improve the simulation, or if now you're moving on to something else.
At this stage, we are okay with this timing. Okay, so in our future work, we are trying to merge with some neural coding things also, and try to see how much time it is taking. If it takes too much time in future, yeah, we have some plan to reduce it, but not model reformulation point of view. Maybe the change in coding things like parallel algorithm or something else. But yeah, from perspective of changing model, yeah, this is our model. We don't have any plan to change anything in our model for reducing the time. Yeah, if we need to change the time, we need to optimize our code, something like that. Yeah.
Okay.
Does it make any sense? Sorry.
Yeah, absolutely. Absolutely. I was just, I mean, like, whenever I have it kind of seemed like a little bit of an open-ended, like, we need to improve it, but how much do we need to improve it? And when do we stop improving it kind of thing?
Yeah.
Like, at what point enough is enough? And I mean, the work that you did clearly made an incredible difference. And I mean, maybe that's enough. Maybe that's all you need to do.
Yeah. Let's see what happens in future, because sometimes when we apply the whole things for real robot, okay, it takes too much time in control thing. So at that stage, I have no idea what will happen in future, but yes, it is almost taking lots of small time. Okay, but yeah, let's see what happens in future. Yeah.
Okay. All right. Thank you. Yeah.
Yeah.
Okay. We have approximately 6 minutes left for questions, and then I believe we have a 20-minute break. Yes, Will.
Hey, I actually have a question about the shaker, number 2. So it was a fun talk with great videos. I'm just curious, because there was the video of the stuffed rat on top of the shaker getting shaken very, very much. Is there actually an intent to shake the animals that much or a demonstration of what the mechanical capabilities are of the system?
No, this was just a demonstration. We cannot use it that way with the animals. So we're just using one single perturbation, vertical, horizontal, like in cranial or caudal or lateral. But we can combine different motions. And maybe in futures or maybe in next years, we will try to combine different actuations. But like the shaker in your video is not possible. Yes, it's going to fly away.
I figured, just thought I'd double check.
Okay. Yeah.
Along those lines, I was kind of as Will, as you were talking about it, I was kind of curious if the forces and accelerations are just like you just wanted to kind of have something that could accomplish or that could reach those types of like very almost like not, I don't want to say violent, but very intense motions. Was that part of the reason for kind of taking it to that level?
Because of we need very short the rats and very small animals, they have very short contact times. So we need the rats at about 0.2, 0.15. The same have small birds that we would like to have in the shaker. And that's why we need about 10 hertz, 10 times this time in order to make perturbations that are really significant to the animals. And that's why we need about 10 hertz. And the case of the force plates that we have mentioned, that we use ITI sensors. So in order to have the ground reaction forces, we measure ground reaction forces due to the motions first without the animal, and we take that out from the motion that or the forces that we measure with the animal in the body.
I see. Okay, that makes sense. Yeah, thank you. Okay, we have a few more minutes if there are any other questions for the short talks. Will, did you have another question, or did you just?
No, that's still the same hand from before.
Okay. Okay. Well, I guess if there are no more questions, we can go ahead and break. And let me bring up the schedule for the rest of today. So we break from 1640 to 17 UTC, and then we start off with our second round of 20-minute talks. So I guess we'll see you all back at 17 UTC.
Great, thank you.
Hi everybody. This is the last section of today. So can you hear me? Yes. Okay. Thank you. So the first talk will be given by Chloe Kegui on the direct assembly and tuning of dynamical neural networks for kinematics. Chloe, you can start now.
Hi, which screen can you see? I think it's the wrong one.
I can see the blue screen. Yeah.
There we go.
That's
All right, is that good?
It's fine now. Yeah.
Okay.
Fine now.
So, my name is Chloe Guy, and I will be giving a presentation about mine and Nick's work relating to dynamical neural networks for kinematics. So whether in an animal or a robot, they need to calculate higher order quantities like the position of the hand in space or the position of foot in space. So in this image, we can see an arm that contains 7 degrees of freedom, and calculating its forward kinematics would require the multiplication of 8 matrices, which is very complicated to formulate and requires computing power. However, even tiny insects can solve this problem. So we look to them for inspiration on how to calculate the forward kinematics of a limb. So insects have non-spiking interneurons in their thoracic ganglia that may enable them to calculate leg kinematics. And these neurons have large branch dendrites throughout the ganglion, along which they receive inputs from many proprioceptors across the leg. By combining all of these separate measurements, their voltage reflects the motion of the leg. For example, the anterior posterior position of the foot, as shown here in the insect. So in robotics, deep neural networks have been trained to compute complicated nonlinear forward kinematics equations. And these have a few drawbacks, including that they are computationally expensive to tune, and they may not model the structure of the peripheral nervous system. So it is hard to relate the structure and tuning of deep neural networks to the structure and tuning of the nervous system, which means this may not be the best tool for our research. Non-spiking interneurons in the thoracic ganglia or the ventral cord may aid in the calculation of task level quantities for motor control. And the question that we seek to answer in this research is, can non-spiking interneurons in the nervous system calculate abstract quantities? This would include combining the angle of multiple leg joints to calculate the position of the foot relative to the body. So the goal of our research is to imply what is known about how insects sense leg joint angles, such as using range fractionated sensory neurons to construct a biologically plausible model of how task level quantities may be computed by the nervous system. In our model, spiking afferents from 2 joints in a leg are integrated by 1 non-spiking interneuron. This is modeled with many dendritic compartments and 1 somatic compartment. The voltage of the somatic compartment encodes the x-coordinate of the foot's position relative to the body. And the conductances between the compartments are tuned using the functional subnetwork approach. So here we have an image of, if you look at the top of the figure A, it shows the schematic of a planar leg with a hip joint and a knee, where each segment is 10 centimeters long. The angle of rotation to the global real axis is defined by theta hip and theta knee. And each joint rotates between 0.75 radians and negative 0.75 radians. So then on the bottom of the figure, which is labeled as B, is a schematic of the sensory neurons from the hip, which are the yellow circles on the left. I don't know if you can see my mouse, but they are these yellow circles here, and the knee, which are the green circles here. And one of each of the hip and knee sensory neurons excites the NSI dendrite compartments in its row or column respectively. every NSI dendrite compartment, which are these smaller blue circles here in the middle, then connect to the NSI soma compartment, or this large blue circle here to the right, creating a unique conductance value that represents the diameter of the bell curves. Oh, sorry, let me go back real quick. And these are the bell curves for the hip neurons, and these are the bell curves for the knee neurons. So the neuron model is designed using the synthetic nervous system, which captures biological details. It runs pretty quickly, and it can be implemented as a robot control system. And the NSI model is constructed from leaky integrator compartments. It's modeled as 25 low capacity dendrite compartments that connect to one high capacity soma compartment, and each 25 are modeled as leaky integrators with a dynamics seen in this equation here. And I just have the variables listed out. I won't go through all of those, just so you can see them. So in the past, we came up with the functional subnetwork approach for tuning, and the goal was to relate network functionality to tuning as directly as possible. So this was done by expressing the gain of a synapse in terms of physiological parameters. For example, the gain of a synapse depends on its conductance with the receiving neuron. And by gain, we mean the ratio between the receiving neuron's voltage and the sending neuron's voltage. So this model, we used ground truth data as gain, and then used that to calculate the conductance between compartments. So here we have two surface plots. On the left is a surface plot of the lateral foot position versus the knee and hip joint angles. And this K equation here corresponds to this X value here. And on the right is a surface plot of the conductance from the dendrite to the soma versus the knee and hip joint angle. And this G equation corresponds to this G value here. So this leads to our results. On the top is a plot of the hip and knee angles versus time, as seen here. And on the bottom is a plot of the position of the foot and the neural representation versus time. The neural representation has been advanced in time as calculated by cross correlation between curves. And we wanted to show, we wanted to sample different combinations of joint angles and see how the system performed. And we wanted to better understand how different parameters affect the accuracy, which leads to the next slide of results. So the first parameter we tested was the width of each sensory neuron's encoding curve. On the left is a plot of the bell curves that relate each cell's injected current to the joint angle with different widths. And as you can see, the dashed blue line shows the bell curve with a width of 15, while the solid orange line shows the bell curve with the width narrowed to 30. And then on the right, we have the plot of the position of the foot and the neural representation versus time with the width set to C equals 30 as seen here in the bell curve comparison. And the time constant is just set to 600 milliseconds, which is the basic time constant that we had. And as you can see here, that narrowing the width of the bell curve causes a decrease in the neural representation's amplitude, seen here. So then we have a plot of the bell curves once again, but this time the solid orange line represents the bell curve that has been widened to 7.5. So then we plotted that, as you can see on the right here, it's the position of the foot and the neural representation versus time, but now with that width of 7.5. And by widening this width of this bell curve, the neural representation's amplitude decreases and the mean value is shifted upwards. So the accuracy of the network encoding also depends on the time constant of the compartments within the model. This is a plot of the position of the foot and the neural representation versus the time with the width equal to 15, which is the basic value that works the best. And the soma compartment's time constant increased by an order of magnitude to 6,000 milliseconds. And you can see here that by lengthening the time constant, the neural representation loses amplitude and lags behind the foot position. Then we have a plot of the position of the foot and the neural representation versus time with the width of 15 once again, and the SOMA's time constant set to 1 millisecond. And this shows by shortening the time constant, the neural representation fluctuates pretty sharply. So then based on these tests, we found the parameter values such that the simulated neuron encodes foot position most accurately. If the sensory coding is too narrow or too wide, neural representation's error increases and the accuracy decreases. And if the time constant is too small, the signal is too variable. And if it's too high, the neural representation lags the leg motion. In the figure, we will more thoroughly explore how to, or in the future, sorry, we will more thoroughly explore how to tune this network to encode the foot position most accurately. So we discussed that higher order quantities may be calculated by the nervous system through the use of many local measurements. We believe local non-spiking interneurons of insects help calculate different quantities for motor control. And we would plan to investigate how the joint's range of motion and the accuracy of network encoding depend on the number of sensory neurons. Range fractionated sensory inputs impinge on the non-spiking interneurons in the leg control network and may support the calculation of higher order quantities. This system was modeled using the synthetic nervous system philosophy to create and tune a dynamical neural network. The performance of the network depends on the Gaussian function width of C and the time constant of the soma tau membrane. And we found parameter values for which the neural representation can follow the path of the position of the foot very closely. Our model was simplified to 5 sensory neurons for both the hip angle and the knee angle. But in reality, insect joint sensors have many more sensory neurons. Also, the network only calculates forward kinematics. So we plan to build networks that calculate other quantities. And we plan to construct models of networks that incorporate whole limb and whole body feedback for both leg local control and ascending sensory signals to the brain. So more recently, I've also been working to create a multi-compartment model of the dendrite of a neuron that will accurately measure the position of the leg based on noisy, redundant sensory input, even if some branches are eliminated. To do so, we connected the dendrite compartments in one direction, as seen in this image here. They go across to the soma. We used constrained optimization to tune the list of intercompartment conductance values called G. Then we set the random values within the G matrix to 0 to explore how resilient the neuron's encoding is. And so the encoding was resilient when small G values were set to 0, as seen on in the middle figure. But it didn't work as well when major connections in the system were set to 0. And I believe this is because I need a completely connected graph. So I would like to maybe try that in the future. But finally, I have been working on updating this project to be able to complete different calculations, such as the X position of the foot, the Y position of the foot, the leg angle, and the leg length. And finally, I would just like to thank the NSF NeuroNext program and CRCNS program for funding this research. And that's all I have.
Thanks, Chloe, for your presentation. It's really wonderful. So I think William has some question. Yeah. So
Hi, Chloe. That was really interesting. Great talk. I just have kind of just a basic kind of fundamental question, which is that, you know, in your work, we're taking the idea that, you know, animal nervous systems, and in particular in this example, in these kind of joint angle kinds of sensing strategies, we break up an analog measurement across multiple sensory neurons, right? So we're representing the angle quantity as multiple different neural activations. And so because you have all those neural activations, you're able to do this matrix strategy. I guess my question is, have you looked at, what advantage you get from doing it from essentially, do you get an advantage by splitting up the angle across multiple neurons and doing it that way? Or could you implement a similar system just with, one neuron that encodes the angle range for the hip and one neuron that encodes the knee? Does that make sense?
Yeah, I think I understand what you're asking. So currently with my, let me see if I can go back. With this research that I've been working on, we are kind of having that problem where splitting it up makes it harder to solve, I guess is what I would say. So maybe trying the one neuron for the hip and the one neuron for the knee would work. I haven't really thought about that.
Okay, and that's fair. That's a fair answer. And I guess, I would partially also think you get more, when you have more dynamics to play with from more compartments, it makes the computation probably a little bit easier. Just wondering if it was something to consider for future.
Yeah. Thank you.
Yeah, Will, can I jump in on that too?
Absolutely.
Yeah, so I think, everything Chloe said is perfectly right. That, this does take some finessing, as evidenced by, for example, you change the sensory coding curves and then it all kind of gets worse. But what I think is potentially so powerful about it is now this is a network that's basically just doing a bunch of addition, and the result is like matrix multiplication. And that to me is a pretty cool power up, if that makes sense. So I'm not saying everything has to be done this way, but I mean, basically it becomes like a lookup table, right? And now you have a lookup table that can calculate any nonlinear weird function you want, and you don't have to worry about having a DNN with a whole bunch of hidden layers where you're trying to map from two analog values to some third nonlinear thing. So that's just my perspective. And, this is all new. So, these are great comments. Thank you.
Thanks.
Hi, is there any question from Vicky's group?
Yeah, it's actually from Michael.
Yeah.
I thought this was really cool, and I think it'd be an exciting thing to put into different robot controllers. And my question is, do you know, or are you looking at whether or not this is kind of a unique mapping? Are there multiple foot positions that, or I guess, are there multiple, foot positions that could map to different hip and knee angle inputs? Basically, can you get a unique value on the voltage of the soma for each input that you're putting in, or do you have to worry about there being some like overlap between commands you'd be sending to, for in this example, a robot leg?
That is a good question.
Do you mind if I offer a thought, Chloe?
No, that would be great.
Michael, that's a great question. My thought is based on what Chloe's done so far. She's been focusing on forward kinematics, which I think is nice because you don't specifically have that problem, I don't think, right? You pick your inputs, you're going to get an output. Even if two inputs give you the same output, that's not necessarily a problem. Now, if we wanted to use this network as part of an inverse kinematics solver, then I think we might run into what you're describing, where you might say, I need a leg height of something, but there are multiple solutions to achieve that. Is that what you're getting at?
Yeah.
Yeah. So in that case, I don't know. I mean, that's kind of what we've been. That's the direction we're headed. So when we run into problems, we'll tell you all about it.
Gotcha. Great. And I guess kind of a different question, but you were looking at the time constant of the soma, and you saw that was affecting the accuracy. Did you look at the input frequencies? So if you had a constant, the same time constant, and you varied the frequency of the way the light was moving, did you see different levels of accuracy or noise?
Yes. Yeah. Oh, I mean, no, I'm sorry. No.
We didn't look at that explicitly.
We didn't look at that. Yes.
Gotcha, okay.
But
Yeah. Gotcha.
But that would be great to do.
Yeah.
Awesome. Thanks so much.
Yeah. So, yeah, I think all the questions have been done for this talk. Yeah. So is it okay to go forward to next talk? So yeah, the next talk is on synthetic nervous system with coupled oscillator controls peristaltic locomotion by Shen Riddle. So yeah, you can start. Yeah.
Share my screen. Hopefully it's the right one. All right. There we go. Should be this one. All right. Are you seeing the presentation properly?
Yeah, it is showing. Yeah, it is visible. Yeah.
All right. My name is Shane. I work with Will Zujun Yu, Dr. Thomas, and Dr. Quinn at Case Western Reserve University. And I'm going to be presenting, I will be presenting the work for a synthetic nervous system using coupled oscillators to control the peristaltic locomotion of a worm robot model. This is funded by NSF, NeuroNex, and NIH to some degree, and we were based out of the Biologically Inspired Robotics Laboratory at Case Western Reserve. So the outline of the presentation, I will first go over the robots themselves and the kinematic model we're using to approximate them. The neuron, synapse, CPG, and sensor models used in the synthetic nervous system, as well as the nervous system itself, and then how we go about tuning that using the functional subnetwork approach, and then what we get out of it in terms of results when we apply the nervous system controller to the kinematic model. And finally, we did a stability analysis because we were interested in making sure that our system was actually a stable, periodic function and not something that was going to just run away on us at some point. So first up is the worm robot kinematic model. Our group has been working on worm robots for over a decade now. One of the earlier ones from Alex Baxter-Baum and Andy Hortzler. You can see this is peristaltic locomotion of a worm. It looks kind of like a worm. On the back, it has, I believe, 1 degree of freedom because on the back there is a cam that operates this large network of cables that causes this motion, but it is ultimately just one motor that controls everything. As we continue, we have been making them softer and softer. So while that robot was soft-bodied, it was being driven by a large and very rigid object at the back of the end of the robot. We have moved on to what is known as CMM worm, compliant modular mesh. It's on the next slide. It is modular in that it is, you can actually kind of see a thumbnail right here. It's modular, and essentially what it does is it isolates this down so that we no longer have this large wheel on the back, this large cam system. We instead have individual motors for individual segments of the robot. And then we've taken this a step further by removing, you can't see it quite yet, but I'll show you in the next slide again. There are plastic vertex pieces that hold the structure together. Removing those entirely to make this now a fully soft structure, we have fabric worm. Instead of those vertex pieces, we just have channels that are woven into this skin, and that gives the robot its structure without having to rely on hard plastic pieces. And most recently, we also have the worm robot that I've been working on, which is the liquid crystal elastomer worm robot. And what happens, it might be a little bit hard to see because it's kind of small, but we have liquid crystal elastomer rings, which exhibit shape memory in 2 directions, two-way shape memory. And when you heat it up, it shrinks down to its memorized state. And when you cool it down, it will return to the other state at which it started. And in this way, we can kind of get the contraction and expansion that you would see from a worm robot
and it can be programmed to move in a peristaltic downwards, but it is entirely soft-bodied and soft actuated. The only hard components on it anymore are the battery power supply and the controller. As promised, on the next slide, we have the CMM work. If I skip up here, you can see the lovely geometric approximation. I'll let it run. Hopefully not sound. All right, I apologize for the sound. I don't know, I guess I can't mute it, but why is this doing? Anyway, it seems to be going to take too much time to play that entire thing. But anyway, the earthworms are hydrostats and they locomote via peristalsis. Our earthworms use a geometric coupled motion to approximate this because it is a little bit difficult to fill a robot up with water and try to compress it properly as an earthworm might. Because hydrostasis is essentially you keep the same volume, and when you change one dimension, another dimension has to change to accommodate that to continue maintain that same volume. So when the radius of a cylinder, for instance, contracts, the cylinder elongates to maintain the same volume. In a similar manner, we have these rhombus approximations. These rhombuses, when you press this, when you pull these horizontal corners towards each other, they will expand outwards, as you see from A to B, and then going the opposite direction, pulling those back the other way, causes them to shrink once more. So going back and forth between this, you can kind of see how you might get a peristaltic locomotion in 2 dimensions rather than 3. For all the robots that we have previously, they all work on a very similar approximation of this. They simply wrap it around a cylinder, wrapped around in a cylindrical shape, but they use the same geometric approximation of the rhombus shapes. All right, now that we've done that, we can move on to the neural models and sensor models. A bit of the background of the synthetic nervous system. I'm sure you've heard quite a bit of the synthetic nervous system talks throughout the day if you've been through the entire conference today. But just a quick overview, synthetic nervous systems are dynamical models of animal nervous systems, and we can directly tune them to perform things such as addition, multiplication, differentiation, using analytical techniques to identify the constraints. These addition, subtraction, multiplication networks are called subnetworks. We can also make different kinds of subnetworks. For instance, we have a CPG subnetwork in the image to the left. And then we can kind of put them all together like pieces of a puzzle and connect them via synapses, maybe some interneurons if we need to. And we can make large networks out of them without fully retuning interior network, depending. The model that we are using for our neurons is, again, the non-spiking leaky integrator model. The synaptic connections between them are transmission synapses. We are approximating the synaptic conductance curve, which is typically a sigmoid that is monotonic and saturates. We're replacing that with a approximation, which is simply a piecewise, a continuous piecewise linear function. This gets enough of the picture across for our system, and it's a lot easier for us to for this simple network. So for other forms of this, and if you're wondering what the U's and the R's are everywhere, we normalize the potential down to start at 0, so we subtract out the resting potential, and we use R for the synaptic potential range. We do this so that we can more easily tune the system without having a lot of things cancel out if we do it this way. It just makes things a little bit nicer. So going to the central pattern generator and sensor models, the central pattern generator uses the same model as the other neurons, except for the CPG neurons, we also incorporate a persistent sodium channel. This enables the temporal dynamics that we need for the central pattern generator to continuously produce a periodic output with the lack of a periodic input. So we use the M and H gates, something similar for the Hodgkin-Huxley model, which you can see over here, but we can ignore the M gate, the M gate time constant simply because it is so much faster than the H gate. When we put this into our equation, we do it as such. There's a sodium current and the conductance over here. You can see the H gates will limit the current conductance.
Yep, just kind of throw it in. And lastly, we have a sensor model. So, I mean, a lot of muscles and neural networks, sorry, a lot of biological nerve systems have proprioception. I think most of them, all of them do. Anyway, proprioception, and in this case, I wanted to kind of simulate muscle fiber spindles in that I wanted to basically enable stretch sensing in the robot. So see how far the worm segment has expanded and give it a form of crude proprioception. When we throw this all together in the synthetic nervous system controller, we end up with this. And I have on the next slide a verbal walkthrough, but I believe it is easier for me to just kind of show you, starting with segment 2, because segment 1 has some, the first part of segment 1 is a little bit iffy because you have to jump start it. But anyway, the first consistent actuation after you start the program, when segment 1 is fully contracted and the neurons and the CPG switch, the expansion neurons become activated. This is simply a half-center oscillator, so it is 2 neurons that will mutually inhibit each other. So one will have a high activation, one will have a low activation, and then due to the sodium conductance, they will eventually switch, depending on, and the timing depends on a couple of the parameters you set. But anyway, when it switches, the expansion neuron becomes high, and the robot starts to re-expand. It sends a signal to the model actuator, and it starts to expand. And when it does, this also excites the U4 interneuron, which then inhibits the U3 interneuron. The U3 interneuron is the command neuron that pauses and restarts the contraction expansion CPG neuron by inhibiting the low neuron and exciting the high neuron to kind of push them away from the thresholds that they need to reach to cause the oscillatory behavior. When it does this, that releases the CPG. So while this one is expanding, this next segment can start contracting. And then you have, eventually when this one goes on to the next step, it will also do the same to the next segment, and so on and so on and so on. So in this way, the oscillators are coupled. When this expansion neuron reaches what we have defined as the physical maximum for the robot, the maximum expansion of the robot, the stretch sensor will send a signal to the interneuron to re-excite it, and then that will then pause this and wait for the next wave to come back. Skipping through this. So now we go to how do we tune this? So the controller is split into n subnetworks, right, as the number of body segments. There are 4 neurons per segment, so you have n times 4 neurons. You can solve for the conductance in this situation at postsynaptic steady state configurations most easily, which the equation we use for that is here, where U star indicates the steady state potential of the postsynaptic neuron. And for an example scenario also, and if you go to the previous slide, or if you go to the verbal diagram, we see this this point in the process where the U4 interneuron is inhibiting the U3 command neuron while the sensor is active, and to tune the system such that U3 is inhibited when U4 is active while the sensor is active as well. We can solve for the synaptic conductance in this particular instance, the synaptic conductance that allows this behavior in this particular instance. Again, we can use this method to tune the interneurons here, but we need to use a different method for the contraction and expansion CPG neuron. We need to do this because the CPG neuron, if either of them goes down to 0 for the minimum potential, the mutual inhibition will cease because there is no longer going to be a signal sent. And if anything that is not a positive signal sent through a transmission synapse will not impact the postsynaptic neuron. So instead we use a small number delta, which for this network, we use a small value about 0.01, just above 0, to keep it out there. And we substitute this and the sodium conductance in the previous equation to solve for the CPG synaptic conductance, which you see down here. Again, so that means we have this and this. There is no synaptic connection rate here between a stretch sensor. This is simply an injected current. And so we see that this system is somewhat tuned now. And when we apply this to the kinematic model we saw before, we get this lovely result. The robot, if it plays, there we go. So the robot will now have a contraction that moves its way down the body in a predictable and controlled manner. And this is, we have achieved peristalsis in this particular situation. It does look like it stops a bit. It doesn't. It's just because the proportional controller gets a little bit slow at the end. But this can be fixed a little bit more via tuning further on. So what do these results actually mean? If you look up here, we have a color-coded version of this chart. This is a 3-segment because 6 is way too, it just got too cluttered in the presentation. So showing you 3 of the segment, a 3-segment robot instead. We have this lovely little wave that kind of follows down. These are the heights of the worm segments at a given time. You can see that segment 1 contracts, and then segment 2 contracts, and then segment 3 contracts, and then the pattern repeats itself. Segment 2's contraction, you can see, is related to the U1 neuron, which is the contraction neuron. That activation causes this contraction, and then when it wants to go back, and then when it goes back to expand, you see that the expansion neuron is now at an elevated state, and the contraction neuron is low. For a visual representation of this, you can see at this time we have the height is high for 1, low for 2, and then for 3 it is at its maximum. But yeah, this is oscillatory behavior, and the CPGs are displaying the proper catch and release type oscillation, and the oscillation is propagated. So we want to do an analyze the stability of this system. So we performed a direct flow K multiplier analysis and approximated the monodromy matrix using direct perturbation of the system. Essentially, what we did is we took the state of the system for the parameters we were interested in, called X naught, which we obtained after letting the system run for a little bit, so it would stabilize entirely. Then we applied the perturbation, which is a very small value, sigma, approximately 0.001. It might have been 0.0001, one of those two. But then we went and let it run for a whole period. In this case, it was for the 6-segment robot that we analyzed, it was 5,220 milliseconds, I believe. And then we evaluated the difference and we performed the monodromy matrix this way. And then we did an eigenvalue estimation. So the eigenvalues, or eigenvalue solution. So the eigenvalues of the monodromy matrix are known as flow K multipliers, and as long as they begin with 1 and decrease in value, the system is considered stable. And when we did this in the direct perturbation of the system, we ended up with an analysis that indicates stability. 0.9945 was our first eigenvalue, which is pretty close to 1 for a simulation. And then it decreased onwards. There are technically 42, as you can see up here, 42 variables, because there were at 6 segments, 6 times 4 plus a couple of things. Essentially ended up with 42 variables, but anything past 3 was essentially 0 and considered trivial. So these 3 main ones, they all just reduced down. It just kept getting smaller and smaller, and it was not practical to kind of put them on this slide because there were 42 of them. But yeah, so we have a stable system. It produced peristalsis in the worm robot model. But for future work, we would like to make the controller more realistic and maybe even apply it to our physical robot. In order to do that, we need to at least make the model a little bit more realistic and make it 3-dimensional. We have someone else in our lab has been working on a different robot, but he has a model that should be adaptable to this situation. And it would allow us to much better represent the motion of the robot, especially the softer LCE robot. But we also need to add kinetics to account for friction and mechanical coupling forces on the structure. We currently don't, as you saw in the you saw before, and you will see at the last slide. The robot tends to slide a bit back and forth because of the way I anchored it, or the model tends to slide a bit back and forth because of the way I anchored it. The friction is necessary for worm robots specifically because if it's too low, then the robot can't push itself forward. And if it's too low, or if it's too high, and if it's too low, then it kind of just twitches in place because it can't get any purchase. Yeah, so those are the adjustments we'd like to make. And then if all goes well, that should be able to be put on one of the physical robots that we have. This is the publications page that I have. And I would like to thank the NSF NeuroNext, NSF Pyre, NeuroNext, and NIH Institute or NIH entities for sponsoring some of this work. And if you have any questions, I'm here.
Yeah, thank you, Singh, for your talk. So, do we have any question? We have 2 minutes, so we can discuss something. So I have a question. Actually, I have 2 questions. Can I ask? Yeah. So in your stability analysis, may I know what is the size of your matrix? So
The
means
monodromy matrix is a 42 by 42 matrix. So if it was a single segment, it would be a 7 by 7. Because we analyzed it for a 6-segment robot, it is a 6 times 7 by 6 times 7, so which is 42 by 42.
okay. So you have used forward Euler method in some part of your computation, right?
Yes.
So
yes.
I felt that it is sometimes very unstable kind of things, but have you observed any kind of issues for using forward Euler method, or it is absolutely fine with your system?
So I did not experience any issues using the forward Euler method for this particular analysis or for this particular simulation. It's typically what a lot of us use for the synthetic nervous systems. Just the dynamic networks. It's one of the easier one of the easier dynamic approximations, simulations. But yeah, so it worked. So I didn't try to go for anything more complicated.
Yeah, it's good. It's good.
There's a lot of other things that go on that can break this system. If that worked, I was happy.
Yeah. And I have seen that you have used liquid crystal elastometer modeling.
Yes.
So in the elasticity modeling, do you use your own theory, or you are following some liquid crystal elasticity theory in your model? So can you please let me know? Yeah.
It's not so much modeling the liquid crystal elastomer as it is using the liquid crystal elastomer to actuate the robot. So it actuates in very much the same way as the motors would, whereby the motors for the robots that we have, like the CMM-ware motor, it simply is a servo that will spool in a wire, and that wire is connected to the circumference of the robot. When the motor spools it in, that circumference will shrink. When it spools it out, that circumference will re-expand. The liquid crystal elastomer is doing that, except using heat, and it just uses its latent shape memory properties to do that.
Yeah.
So it will just shrink and expand. I'm not modeling the elastomer itself. I am simply modeling the expansion and contraction nature of the peristaltic motion for this geometry.
Yeah, thank you. So I think Falk has some question.
Yeah, thank you for the nice talk. And I have a follow-up question on that, actually.
Okay.
Because you just said that you only are partially simulating it. So could you actually translate this system to, for example, a peristaltic pumping system or something? So the simulation, because there we also have just the peristalsis motion. Or would that be actually hard to do that? That's the question I have.
It would depend on the kinematics of the peristaltic pump. So this is particularly set up such that there's an oscillator that drives what is essentially between each of these. Let me just point it out. Between each of these vertical and these two vertical vertices, there was modeled a linear actuator that was controlled by this. It's essentially it was essentially the easiest way to go about a shrinking and contraction of a string or of the LCE in a linear fashion in 2 dimensions. So just kind of narrowing it down from a 3-dimensional cylinder into because the robots that we have also been modeled using the rhombuses in this way, but instead of trying to model it in 3 dimensions initially, the easiest way to start was doing this. But anyway, this model is based upon there being these linear actuators between here. So if there would have to probably be some retuning if for a different actuation system for a peristaltic pump. But I believe that it would be doable to do something like this. Yes.
Yeah, cool. Thanks. Yeah, just interested because I developed a peristaltic pumping system based on pneumatics, where we had pneumatic cushions actually extending like your linear actuator, but we had two cushions going inward and not one contracting. But it's quite interesting. Yeah, thanks.
Yeah, that actually might be fairly easy to adapt then, depending
Yeah,
on.
I think so too. Then yeah, let's talk maybe later or in the next days. Cool. Thanks, man.
Yep. Thank you.
Yeah. So we can go forward to next talk. So the next talk is on 10-year Living Machines Conference. So our speaker is Theophile Corniel.
Yes,
you can
hi.
start. Yeah. Thank
Okay,
you.
so I'm going to share my screen now.
Yeah, of course.
This should be okay, I believe.
Yeah, it's absolutely fine. Yeah.
Right. So hello, everyone. My name is Theophile Corniel, and I'm going to present our article titled 10 Years of Living Machines Conferences, Transformers-Based Automated Topic Grouping. So in this work, we endeavor to study the evolution of the International Conference on Biomimetic and Biohybrid Systems, with its first edition taking place in July 2012. It's an interdisciplinary conference that lies at the intersection of a number of scientific fields, such as life sciences, engineering, physics, chemistry, computer science, and a bunch of other fields. So interdisciplinary research is obviously great and very promising, but it also comes with its share of difficulties, notably the fact that it draws from and creates a very heterogeneous literature that requires the high level understanding of a number of different fields. So this is actually a problem. First of all, it's a high barrier of entry to, well, this conference, but more generally interdisciplinary research. And it's becoming more and more of a problem as there is more and more scientific production that's happening every day in the world. So how can we basically create tools to better understand and analyze this conference as an example, but more generally scientific production? We decided to try and use the Living Machines Conference as it was, as we said, very interdisciplinary, and it's still a fairly small number of articles that are published in order to develop a methodology to read, classify, and analyze the roughly 500 articles that were published in the conference proceedings. So the goal is to have a methodology to automatically analyze and characterize the different research themes in the conference, and to be able to understand how it intersects with other fields that are closely related, such as soft robotics or biomimetics. To perform this analysis, well, it's basically in the field of bibliometric analysis. So there are, of course, existing works and a number of candidate methods that I'm just going to present very briefly potential candidates here, but we can either use article keywords, so keywords given by the authors to describe some of the contents of their articles here. Co-citation networks are also something that we find pretty often in bibliometric analysis. And finally, we see more and more NLP-based methods that use artificial intelligence, to directly analyze the contents of the articles. Each of them has some advantages and drawbacks, obviously, but we chose to go with the NLP-based method as it directly analyzes the contents of the article, so the raw text, and is more versatile, we believe, in order to create meaningful thematic clusters. And so once we've said that, how do we actually create the clusters? Well, first of all, we need our data sets. So as I said, we are going to study the Living Machines Conference proceedings. There's a total of 494 articles, and we only keep 461 of them after curation, where we remove articles that are not, where we don't have complete information, the title, the abstract, and the author list. And we perform light pre-processing on the text itself. So we keep all text as we're using transformers-based architectures, but we transform it into lowercase characters and remove all non-alphabetical characters as well. So that's our data set. So for each article, we we get the text of the article by concatenating the title and the abstract of the article. So we don't work with the core for a number of reasons that I'm not going to describe much here, but basically, as the articles can be written in very different manners, it's easier at least at this moment to use concentrated information such as is present in the abstract and the title. So the methodology is as follows. We use the text of each article to create a latent representation using a Cybert model. So the latent representation is a high-dimensional representation, a high-dimensional numerical vector that basically projects the text of the article into a latent space. The main thing to understand is that basically latent space has the feature that two articles that are in proximity in the latent space have similar themes. Then we perform dimensionality reduction steps to lower the dimensional representations of our articles for technical reasons, such as reducing the load, the computational load, and compressing the information for distance calculations. And finally, we perform clustering of articles by their themes using HDB scan. So what I'm going to call a cluster here is a group of articles that are determined by the algorithm to have similar topics, similar themes of research on a fairly abstract level. So here we follow the methodology is basically following the BERT topic methodology, which was published earlier this year in 2022. The main difference being that we use a Cybert model. So it's a language model that's trained on scientific literature rather than a more general language model. So once we've said all that, it's very theoretical and abstract, I guess, on some levels. So what exactly do we get once the algorithm has performed its job well? Here you have a two-dimensional representation of the latent space, where each dot is an article and each color is the cluster it was allocated to. So here we have 8 clusters from 0 to 7 of varying colors, and we have a -1 cluster, which is gray dots, which actually correspond to unlabeled articles that could not be labeled by the algorithm as they were, as it didn't have a high enough certainty of which cluster the articles belong to. So once we have all these clusters, we obviously want to see if they have some, if they make sense in the way that the clusters actually have understandable themes and differentiated themes between the different clusters here. So to determine the theme of the various clusters, we use a method that we will call representative n-grams, where for each cluster we try and determine the n-grams, so the combination of words that are most disproportionately represented in a cluster compared to the rest of the clusters in our data set. This is done using the class-based TF-IDF methodology that was also presented in the BERT topic paper I mentioned earlier. And so for each cluster, we then get a result that looks like this. So it's basically a list of words that are representative of the cluster itself. So using this list of words and manual analysis of the articles in the various clusters, we labeled the cluster by their themes. So for instance, cluster 0 contains articles dealing with the decentralized locomotion, cluster 1 bipedal locomotion, cluster 3 locomotion, for instance, 5 is about perception that differs from tactile perception, cluster 6 is about learning, and cluster 7 about human robots. To globally summarize, so we have 8 different clusters that have varying size and that have varying themes. But we also see that, well, some of them are obviously quite interlinked, like bipedal locomotion, decentralized locomotion, more general locomotion, obviously have a common overarching theme. Same with tactile and perception, and same are human robotic learning. So basically, we have the three great themes of robot design, where we have behavior clusters, behavior-related clusters, sensing-related clusters, and actuation-related clusters. Furthermore, we have a cluster that deals with soft robotics in general. So with, for instance, materials related, articles or matters that are not directly related to robotics, but are in the same field. And so as we see, we have some clusters that are very different, but some clusters that are also quite similar in theme. So we want to see exactly how they differ and how they are similar. In order to do that, we computed a pairwise cluster normalized cluster similarity matrix, where basically we take, for each pair of clusters, we take all individual articles in the cluster and see the similarity between all the individual articles in cluster 1 and the individual articles in cluster 2. So this gives us a similarity value between the two clusters, and then we min-max normalize this similarity value to get rows where we have the least similar cluster to the row cluster that has similarity value 0, and the most similar has similarity value 1. So for instance, decentralized locomotion here is most similar to decentralized locomotion itself and least similar to human robot. So I'm not going to spend too much time describing this matrix, but basically we see that the locomotion-related clusters are fairly similar to each other and are very dissimilar to behavior-related clusters, and that the sensing-related clusters are kind of in the middle, being similar to, fairly similar to behavior and to locomotion, which could be something that we kind of expect, as sensing draws and deals with both actuation and behavior. Soft robotics, however, is quite a special case, as if we look at the column here, it's both not maximally similar to itself and is very dissimilar to all the rest. So it's really a different field compared to the other fields, to the other themes in the Living Machines conference. So obviously what we also want to do, as the title says, is study the evolution of living machines through the 10 years of its existence. And so we look at the temporal evolution of the proportion of each theme, rather, for each edition of the conference. So here is each edition of the conference, and here is the proportion of articles in each cluster. And on the right, we have an aggregated view of the distribution of the proportion of each cluster for each year. Basically, we don't have a massive temporal evolution with clusters that are themes, rather, that appear or disappear. But we see that learning, for instance, here, and soft robotics here as well, are two themes that are quite largely represented for each year, and that also happen to be the two biggest themes in number of articles in our conference. So this is not such a surprising result. And but so far we do not see major temporal evolution, possibly because, well, first of all, the conference has only started in 2012, so it's a short time frame in terms of scientific publications, and it's also a fairly small amount of papers with less than 500 total articles. So it's hard to have some very specific themes that are still evolving and coming and going with such a short time frame and such a small number of publications. Then we look at the co-authorship graph to see how different people from the different fields collaborate. So basically, we take all co-authors to create a co-authorship network in a fairly straightforward manner, and we each author to the cluster in which he published the most. So we see that we have some large number of small groups on the outside of the graph that are usually quite thematically defined, such as, for instance, the Daniel Goleman and his co-authors here, that all specialize in locomotion-related publications. And we have a number of large components, such as this one here, or this one, that are more interdisciplinary. So they still have some thematic focus, like this cluster here is more about soft robotics. Its authors deal more with soft robotics, and here we have more behavior-related clusters. But we see that there is some collaboration going on with authors from different fields, which is obviously necessary in order to perform research such as the one done in Living Machines conferences. Finally, as we said, we also want to see how the publications in Living Machines differ or are similar to other related fields. So it's a work in progress, but we're studying publications from Soft Robotics Journal and Bay Inspiration Biomimetics Journal as well. And so this is I'm not going to describe the matrix itself because it's a bit too large and not easy to read, but basically what we see when we look in more detail here, it's the same kind of matrix as was shown earlier, but we see that the behavior-related topics in living machines, such as learning or human robot, are actually quite weakly represented in soft robotics and bioinspiration biomimetics. So it's really a kind of specificity of the Living Machines conference that is actually not treated as widely the other corpora. And to finally, so we studied which themes were represented in the Living Machines conference, but we can also ask ourselves which themes remain underrepresented. To determine a list of themes that could use more, that we could study, we looked at the keywords at the plenary talks, rather, of the 2021 edition of the Living Machines conference to select the themes that were seen as prospective fields of study by the presenters. So we see that the most mentioned theme in the articles is the efficiency theme, but it's only mentioned in 8 articles out of 500. So with the second one being energy autonomy in 5 articles, and then we fall to 3 and 2 mentions. This is actually, so yeah, these were seen as works that need further studying and are actually currently underrepresented. So to conclude, we presented a novel methodology to analyze and extract research topics from scientific literature. So no big surprise in that we find themes that are actually bio-inspired and actually correspond to a bio-inspired view of the main themes of robotics. But we did not see any thematic temporal evolution for reasons potentially pertaining to the corpus. But so we still find themes that make sense and that actually could be expected, but are also specific enough to encourage us in working on the methodology, because it does seem to be able to yield results in a fairly homogeneous corpus. So could be also a good tool of study for larger and more diverse corpi. And basically what's next for us is obviously improvement in the methodology, such as keyword generation or clustering. As we've seen, there are some problems with the unlabeled articles, and our inability to properly use complete articles is something that we should work on, but comes with its own set of challenges. And there is also the application of the methodology to other corpora to see what we're getting, such as we've started to do. That's it. Thank you for your attention. And if you have any questions, I'm more than happy to answer.
Thank you. It is really a wonderful topic. So is there any question? Yeah, Roger has a question. Yeah.
I really enjoyed this. This was fun. One thing I noticed in unlabeled, there were like 3 instances of words that had to go with the moth. It was like Manduca sexta and hawk moth, and there were like, you know, 3 instances of the same creature. And there's 3 instances of the same creature. Manduca sexta hawk moth, Manduca sexta hawk moth. Anyway, when you have locomotion, perhaps you could divide it up into flight versus ground locomotion, or flight versus legged locomotion or something, and that would have another cluster, I suppose. But anyway, I really enjoyed this. This is great.
Well, thank you very much. Yeah. So there are a number of ways to improve these representative n-grams, and it's one of the main areas of research, because these can be quite sensitive to small fluctuations in terms of, as we look at the occurrence of a combination of words in a cluster versus the whole, versus the occurrence of the same combination of words in the complete corpus. If the word occurs just a few times in the complete corpus, but also occurs always in the same cluster, the rare few times that they do occur, they're going to have some comparatively high values of representativeness, let's say. So this could be an artifact in the manner that the representative n-grams are shown. But you were mentioning flying, like clusters that are about flight, basically. So not to spoil anything, but yeah, basically depending on the corpus we look, like for instance, the bioinspiration biomimetics corpus, if we do the same analysis, we have a very defined flight cluster. So it depends on the number of articles that are about the topic as well. Here, I guess we just have a few articles that mention Sexta hawkmoth or related topics.
Yeah. So Vicky has some question. Yeah, both of the Vicky. Yeah. So can you start question? Yeah.
The other Vicky was hand raised first, so I'll let her go.
Oh, sorry.
Thank you. To be honest, it's not a question. It's more of a remark that, like, really nice. I think that it's a very nice way to show, first of all, the vast variety of interesting topics that living machines has been, let's say, showcasing the last years. A very good way of actually finishing the first day, that it's after the 10 years of the living machines, right? Very nice, Alex, the selection of this paper. And honestly, this is one thing that I think that we should have done ourselves, like to kind of showcase what you're showcasing. So thank you very much for this. It's a very interesting paper. I'm looking forward to read it all. So just wanted to say that. Really, really nice. That was it. Thank you.
Thank you.
Yeah. You can start, Vicky.
Okay, thank you. Yeah, so this was, I also found this really interesting. And I just had a couple questions about why you think it's classified some things certain ways. I guess the first question is, where did biohybrids end up? I suppose a lot of people would probably guess that I'd ask that. Because I was a little surprised that like Tegote-based control comes out as its own theme and biohybrid does not. And I would like wonder why. And why is stick insect in tactile? Just like, why do you think these things came out this way? I know you did this cool algorithm, the algorithm did this, but do you have any insight into why?
Yeah, so stick insect, I'm gonna answer with the last question actually, but stick insect, it depends on the articles in which stick insect was mentioned. So for instance, I'm missing the words, but I think there's some research that's done on how the stick insects have tactile feedback from their legs. And so if basically the article, when we create the latent representation, will have information on, basically we'll classify this article as being in the tactile sector of the latent space, right? But if stick insect is only mentioned in these papers, in the whole corpus, then it's going to have a higher representativeness score, even though it is not truly representative of the theme itself, right? So it's just like some artifact of these representative n-grams that is actually a limitation of this study as so far. To create these clusters, we still have to perform some relatively exhaustive manual analysis of the articles in each cluster to verify that indeed stick insect is not very interesting in itself, but it's articles that use the stick insect as an example to have to study tactile interfaces with the outside world. So that's why some words here, some are just misclassified, as the algorithm misclassifies some articles as well, sadly, but it can be improved. And some are just not very descriptive, but actually make sense when you look at the rest of the article. It's just partial information here that's not quite satisfactory. And when you mentioned biohybrid, it's I think, and I don't have a straight answer as I haven't asked myself this, but biohybrid is quite large and can be studied in many different ways. So it could be through perception related matters, could be through locomotion related matters, and so on and so forth. So and in a certain way, all the articles in the conference are about biohybridism, if that's the word, biohybrid systems rather. So
I guess I mean specifically like muscle-based biohybrids. So maybe that's where the distinction is. Like all of them say biohybrid because it's the conference, and so the muscle-based ones don't stand out.
So I can add something maybe. In fact, you're right. You're both right. The cluster doesn't appear for biohybrids, and I think for two reasons. It's because what Theo was saying is that it pertains to perception or to moving or to bending on something like that, and there are too few papers for the moment specifically devoted to biohybrid systems. And so they are still diluted. Maybe in the next year or with different corpora, we will see the emergence of the field of biohybrid robots if we extend beyond, let's say, the living conferences. And that's one of the frustrations is we didn't see an evolution, a clear evolution, although we know there are evolution. We know ourselves, because we are expert in the domain, that there are evolution. And biohybrid is one of the emergent field that doesn't appear yet as a cluster because the corpus is too small, and we should extend maybe beyond the living conference and to robotics in general. That's maybe the two reasons.
So what you're saying is my students need to submit more papers.
That's true, always.
So I think Paul has some question. Yeah.
Yeah, so thank you very much for this really interesting study. And as you know, in 2013, right, Nathan Lapora also published a bibliometric analysis of the field of biomimetics. Have you also looked at that paper?
Yeah, so it was kind of so one of the inspirations to do this work as well.
Yeah.
But if I remember the paper correct, yeah.
If you talk about trends, it also might be interesting to look at the kind of clusters that he identified there and how this might coincide with the clusters you have extracted.
Yeah, and one of the main differences, I believe, is that there are methodological differences, but the main one, in my opinion, is that the data sets used on his end are from a number of journals that we so far do not have access to.
Mm-hmm.
And
he's really mapping out anything around biomimetics, right? So he looks at all published papers and then see how can we cluster this relative to this notion of biomimetics. And you took the other approach by saying, no, let's just look at the stuff that's in the living machine conferences, and then we take it from there. But now there's an interesting bridge, right? Because now you would, of course, want to know which other conferences does this then overlap or not, right? How is this situated then in that broader field of biomimetics? So are you thinking about also looking at that?
Yeah, for sure. And one of the, two reasons, I guess, why we chose the Living Machines conference is one, as we said, is so it's a single conference. So getting the data itself was easier than getting data from a number of different papers, of journals rather. And another reason is that it's still fairly small, so we can develop the methodology while still having some, being able to exhaustively manually check our results.
Exactly.
And then,
very good. Yeah.
once this works, and
Yeah.
we're still working on it, but it would be extremely interesting to perform our analysis in the same manner that Nathan Lepora did, so in the same journals, for instance, and see how our clusters found by our
Right.
algorithm rather map to his cluster. And so if they do, that's the result. If they do not, why don't they? And has something
Exactly.
changed, or is it just
Yeah.
methodology? So that's
Now,
something
I
that
would
we
be
hope to
very
do.
happy to follow up on that, because we have another number of, let's say, road mapping challenges for which you would need these kinds of tools in the domain of biomimetics and neurotechnology. So I'll be very happy to discuss that further. So to also
great
test your
pleasure.
pipeline, you know?
Yeah, sure.
So thank you very much. Great work. Thank you.
Thank you.
Yeah. So Alex has written something. Soft robotics people like to talk about what their work presents. So Alex, do you want to add something?
It was just a quip that work present is under cluster 2.
Oh, okay. Yeah. So that's another of the limitations of these kind of representative n-grams, which is work present is quite obviously something that's often written in articles such as, oh, this work presents the elaboration of a new methodology of thematic clustering analysis. And so for some reason, these papers in the soft robotics clusters were more often, I mean, disproportionately had this formulation in them. And so it adds no information whatsoever and is noise more than interesting information, but it's somehow it was more often in soft cluster 2 papers compared to the rest of the other clusters. And so it found itself as an important formulation for cluster 2. But it's, so here we only show the top 5, and for obviously to determine the topics of the clusters, we added some more, we looked at more than 5 n-grams, and that the 1 gram, the 2 grams, the 3 grams. So this is just a small part of the information that we use to present, like to determine the themes of the clusters. And we are still looking for smarter ways to generate the keywords, as there are some, but it's also a trade-off between having some having high performance in the generation of keywords. So we could use, for instance, transformer-based methods in order to find the keywords that are the most similar to the theme of the cluster itself by aggregating all the articles in the cluster and having a way to find a list of keywords that are thematically similar rather than just statistical ways to find the n-grams as we currently do. But the drawback of such deep learning methods, for instance, is that it's more computationally expensive and is more data set specific, as you have to look at the vocabulary of your whole data set. And this one is very cheap and fast, even though it has drawbacks. So for now, this was a satisfactory but not perfect way to find our keywords.
Yeah. So I have one question. I can see that for living machine, the data is almost for 10 years. So may I know for other journals, what is the duration? You may have mentioned it, but I may have missed it. So
No, I
what
did
is
not
that?
mention.
Yeah.
So we've looked at 3 journals so far. So Living Machines has information, has proceedings between 2012 and 2020 included. The Soft Robotics Journal, if I remember correctly, started publishing in 2014. So that's even shorter and less articles. And the Bioinspiration and Biomimetics Journal started publishing in 2006, if I remember correctly. So that's that's low. So you have way more articles in there and a longer time frame. So these are some interesting. So you have two differences. One having less papers and a shorter time frame, another being larger and more longer time frame. So it's also interesting to see how the algorithm behaves in these circumstances as well.
Yeah. So I was feeling very interested about the figure of collaboration kind of thing. On your slide, what the slide number? Yes, this is really interesting. So, okay. So, I can say there are 3 or 4 cluster kind of things. So, and this data is only from living machine, right? Not from,
Yes.
okay. Okay. Yes.
Yeah, so each node here is an individual author, and they are linked if two nodes are linked if the authors published a paper together in the Living Machines conference.
Okay. Okay. So is it possible to check for other journals? Is it possible?
As long as we have the author information for each article, then yeah, we can generate this figure for other journals as well.
Yeah. So I have one very silly question that how did you check that thing, by the author's first name and last name or using ORCID ID? Because sometimes names change.
Okay, so
Okay.
that's a very good question actually, as there's no very easy way to do that because names are notoriously hard to deal with, in my opinion.
Yeah.
Because
Yes.
even in the Living Machines conference, for instance, in the different proceedings, the names are not always written in the same manner. So you can have, for José Alba here, my PhD advisor, he could be José Alba with an accent on the E. It could be José I.Alba. So we did an approximation in that we take the first and last name, and I then run Levenstein distance, which is basically a distance between two chains of characters. And if the distance is small, so the chains of characters are similar, so with a very high similarity level, for instance, 1 letter difference, I would merge the two. So if two people have the same name, it's virtually impossible to distinguish. If they have close names, but not if they have the different orthograph of the same name, then we merge the two and consider them to be the same author. But it becomes very hard when we have some similar surnames and similar first names, and that's a limitation that without the ORCID IDs is very hard to deal with, I think.
Yes. Yes. Yeah. Yeah. Thank you. Thank you. It is last time I want to say this is really interesting presentation, and it's really wonderful work. Yeah.
Thank you very much.
Yeah. So I think I have crossed lots of time. I am very bad in maintaining time. So what to do? So we can say goodbye for today. Is it okay?
Thank you, everybody,
Thank you
for
very
coming
much
in
for your
today.
work.
Yeah. Bye. Thank you. Yeah.
Yeah,
Thank you, bye.
that was the last talk for today. Feel free to hop on Discord. There's voice chats in there as well as text chats if you want to keep some conversations going. And otherwise, we'll see everybody again tomorrow, same time.
Yeah. Bye. Bye.